Hey guys, good evening and welcome back again to your one more amazing session of biology. And for the first time in the history of Unacademy, a chemistry educator is going to teach you the top-notch concepts of biology. Are you ready for it? Someone is writing Ambika sir. Good evening, people. Good evening. So let me know quickly in the chats how excited you guys are for the session. Let me know quickly, people, in the chats. Yeah, I know you must be thinking how he will be able to teach the biology, but don't worry. I'm here. I can teach anything. Yeah, so let me know, people. All ready? Sir, can we trust you in biology? Of course. I'll teach better biology than your Ambika ma'am, okay? I'll teach better biology than your Ambika ma'am. So, do you know the topics which we shall be covering today? The first topic is going to be cell cycle, which I'll be discussing with all of you. Then we'll be talking about mitosis, meiosis, RAAS, sliding filament theory, mechanism of breathing, O2-HB dissociation curve, and ECG. So all these topics I'll be discussing with all of you.
Hi everyone, I hope you all are doing great. Students, I just want to say one thing, right? Wasim sir, if you will tell me the full form of RAS without Google or without the help of any book, I'll resign right now.
What?
If you will tell me the full form of this without cheating, without Google, without any book, I'll resign right now. I challenge you.
See, the point is right now students are in very need of biology, so I don't want you to resign. So let me not give you the correct answer.
Chalo, theek hai. One more question. Just tell me the difference in between physiology and psychology.
Physiology is a nice subject. It is one of your units in your zoology.
No. Somebody was asking me, na, ki, sir, somebody was asking, na, can we trust sir? Can we trust sir for the biology?
Of course.
The answer is no. As a physiology or biology?
What?
Or a psychology? What's the difference?
See, psychology is something related to brain, right? Yeah.
Good.
Physiology is something which is related to the, human body.
Achha, my question is, psychology, is it related to the brain or is it related to the mind? And what is the difference in between brain? You got the answer?
See,
you got the answer?
I think one is thermodynamically controlled and one is kinetically controlled. Now you tell me the answer of, you tell me the difference between case kinetically controlled and thermodynamic.
I wish I could. He got one answer. What is he saying? Thermodynamically controlled or kinetically?
Next chemistry class I'll take on Monday. On Monday I'll take the next chemistry class, okay?
And yes, I will be taking one more session of top 50 concepts because you know that in our case there are a lot of explanations, so I'll be taking it on Sunday.
Yeah, and on Saturday there is a test series coming up for all of you. Okay, yes. I know the last month is very crucial. Perfect, you at least need to write some 20 to 30 full syllabus tests. So for that purpose, on coming Saturday, I'll be launching a test series with all of you. So please do join that particular session as well.
And after so long, you people are watching us together, na?
I know you. I'm pretty much sure you guys must be missing the cat fight, huh?
No, today, now, every day we fight. We fight a lot.
Exactly.
His life was not going well. He called me and he was like, I'll make your life hell as well. His life was literally not going well.
So do you want to study biology right now? Yeah?
Of course.
ECG. I can tell you the full form of ECG.
I'll give you if you will tell me.
Engine. Clutch.
You cannot trust him for the biology at least.
Gear. That's what it keeps on coming in the ads, right? ECG. Engine, clutch, gear. Okay. What else? Oxygen, hemoglobin, dissociation curve.
What is hemoglobin?
It is blood. No, wait. Huh?
Blood. It is blood. No. Please record this part.
Achha, hemoglobin is
blood. Please record this part. I am going to put it in our shorts.
Hemoglobin it's red in color. Huh? Wait, it's red. So it is खून.
It is खून ना? ये खून है भाई, कुछ नहीं हो सकता. मेरी लेक्चर 5-6 बेसिक सीख लो.
Okay.
चलो अब जाओ leave.
Okay.
एक मिनट.
Guys, do you want me to leave?
See, get me a glass of water. नहीं.
I'll get you a bottle of water. Don't worry.
Bottle of water. ठीक.
Hemoglobin is a pigment.
हाँ.
Okay.
किसने लिखा था? किसने लिखा था?
Hemoglobin is a pigment.
ये pigment. Thanks Mr. MBLZ.
The ECG abbreviation of the year. Infant milk year. Right?
But बच्चे, this abbreviation will not give you the marks, ना? चलो sir. Sir, you go. You get me a water bottle. Till then, I'll teach them.
Okay.
So.
Chalo guys.
मेरे को.
Chalo, you carry on, huh?
हाँ.
Okay, I'll keep on coming in between. Keep on coming.
He'll bring a glass of water. He'll bring a cup of tea. Right?
And
Today I am his guest.
exactly.
Guys,
you can trust me in biology. Chill.
No. You cannot.
You can.
People, you cannot. If you really want to protect yourself, then please don't. Please don't.
I'll come in between, okay?
हाँ. So, students.
Or should I be here? I'll watch you.
And what will you do then?
I'll learn the concepts.
And do you know, guys, why I'm here at Srinagar? Do you know? Do you know why? Because our Wasim sir is getting married.
Oh.
Exactly. Our Wasim sir is getting married. That is why I'm here.
No.
Exactly. He
No, guys,
is getting married. That is why I'm here. He's not accepting it, but he is.
He's lying too.
No. He is getting married.
Do you think she'll be taking the sessions at my place if I'm getting married?
She is
would
getting
be busy
married
serving the
within...
guests with the cold drinks and stuff, right?
He's getting married in 15 days. Arey, I'll tell you about her. Don't worry.
अब की बार 400 पार.
अब की बार 400 पार. ठीक है? He is getting married. That's why I'm here. Otherwise, why would?
Someone is saying, who is your angel, sir?
I know.
My angel is,
I'll tell you about her. She's such a beauty. Trust me. Seriously.
Guys, don't listen to her, okay?
It's a fact. He's getting married. That's why I'm here. Otherwise, why would I? Arey, birthday party.
Who's that lucky girl?
She'll be lucky. Seriously, she'll be lucky.
She must be chemistry addicted.
She's not. Trust me, she's not. She loves biology, but... Chalo.
That's why I'm learning biology for some time, I'm trying
Seriously,
to learn
he's
biology.
getting married.
No, she's no. Such बोल यार.
सच बोल रही हूँ यार. मैं सच बोल रही हूँ यार.
No, guys. Nothing like that for now. You'll get to know, okay? My marriage will be live on YouTube. Don't worry. All of you would be invited.
He's
provided all of you would be in the dream comments
like
by
40s.
then. Huh?
He's married like 40s. Okay, बेइज्जती. चलो अब चलते हैं. चलो go.
So मैं पढ़ूंगा यार.
चलो ठीक है student. So students, the first topic that we are going to start is cell cycle.
Or this one.
दिखता नहीं है यहाँ. ठीक. Okay. So students.
No, not this one. Wait.
कुछ और निकल रहा था मुंह से, मैंने बोला नहीं. Cell cycle. Okay. So why are we going to talk about this particular topics? If you will check NEET 2024 paper plus re-NEET 2024, there are 3 questions from this topic only. Right, from this particular chapter only. So cell cycle, mitosis, meiosis, they are the basics of biology. And this time also 3 to 4 questions are expected from these 3 topics, right, bache? So yes, you know that in the case of like in physics and chemistry, we have the direct topic, this question, solve this question, you'll get this much marks. But here we have to complete the topic, right? We have to study the entire topic, then we will be able to get good marks. Okay, so what we have to do, we will finish the cell cycle first, but yeah, it is not as per NCERT. I'll highlight everything. No, to the point, directly as per MCQ, then mitosis and meiosis. Okay? He's just jealous. Come back to this. I am liking this number.
Huh?
You stand here. So first of all, the topic is cell division. So first point is, why do a cell divide?
Wait,
Yes, sir.
I'll change my jacket and come, okay?
हाँ.
You carry on. I'll come in just 2 minutes.
Good. So, speak anything in Tamil, sir. Irukenga. This is what I have learned. And volume increase. Volume is less? Is it so? I believe it's fine. Okay. So, students, the point is cell division. Why do a cell divide? A cell is going to divide when there is the disturbance in surface to volume ratio, number 1. when there is a disturbance in surface to volume ratio. Students, I'm repeating this thing again. Today, I will not highlight everything from the NCERT. As I said, top 50 concepts of biology. And trust me, if you will complete this, if I'm saying top 50 concepts of biology easily, my dear people, easily from these 50 concepts, you people can expect 60 to 70 questions very easily. Trust me on that, very easily. So 70 question means you know that how much marks are going to be there. Okay, right, bache? Okay, chali. So let's get started. So as I said, when it comes to the cell division, surface to volume ratio will get disturbed. And second thing is what? Second thing is the nucleocytoplasmic ratio. When these two things, right, when they get disturbed, the cell is going to divide. Now, what kind of disturbance is there? It's very simple. You talk about a small cell, right? When you talk about a small cell, obviously small in size, it is active cell. It is immature cell. It is small cell, it is active cell, it is immature cell. Now you know that in a small cell, the nucleus size will be large, right? And this nucleus will be able to take over or to control the function. It is capable of controlling the function of this particular cell. Now you can go here. So this small cell, it will grow in size, right? Nucleus size will remain same, but the cytoplasmic growth is there. So because of that, this nucleus is not capable of, right? It is not efficient enough to control the activity of the cell. So basically what is going to happen? The nucleocytoplasmic ratio will get disturbed. The nucleocytoplasmic ratio will get disturbed, right? What is going to happen? The nucleocytoplasmic ratio will get disturbed. All eyes on Wasim sir. So this is going to happen. So this is the first thing. Then is the surface to volume ratio. So you tell me, if a cell is small in size, its surface area is more or its volume? Something
less.
If a cell is small in size, its surface area is less, or
Volume is less.
you tell me, no? I'm asking you.
Volume will be less.
And what about the surface area?
Also will.
Again, I'm asking you the same thing.
Yeah.
If a cell is small in size,
Cell is small in size. Surface area
surface area is more and the volume will be less.
is more.
हाँ, छोटी चीज है, बड़ी चीज में surface area कम हो जाता है.
What? Wait. Just guys, wait. एक मिनट pen दे.
हाँ.
This is a
Read about the
small
surface.
football.
हाँ.
So,
Big
bigger size
football.
football.
हाँ.
Okay. It's voluminous. Yes.
हाँ.
It's voluminous.
More. हाँ.
Right? Okay.
What about surface area?
Surface area of this smaller football. Yes.
नहीं, इसका ज़्यादा है.
Surface area of basic. Okay, you are talking about large number of cells. Right?
I am saying that if a cell is small in size, if something is smaller in size, the surface is...
What is she saying? Yeah.
The surface area is more in that case.
Manu, this is what you study in biology. This is what is asked in biology. I am not going to like it.
See, NEET, in the NEET examination, 360 marks are from biology. They have to study it. They don't have choice.
Wait, yeah.
Yeah, seriously. Guys, in the case of NEET endgame also, or as we have already discussed, after this part, we will start the NEET endgame only.
Guys,
Endgame, no?
what is the formula to calculate surface area of a sphere? What is the formula to calculate surface area of the sphere?
That is correct. Seriously.
Maybe your concepts are different, yeah?
Yeah. You go and you check. I heard somebody saying, please teach JEE chemistry.
No, that is volume. That's volume. I'm talking about the surface area of the sphere. It is 4 pi r square.
Yeah, I'm telling you.
Give me the pen, yeah?
अभी bio की class होने दो.
Guys, this is small sphere. This is bigger size sphere. This has got the radius r. This has got the radius capital R. Its sphere is 4. Its surface area is 4 pi r square. This is 4 pi capital R square. Now capital R is greater than small r. Capital R is greater than small r. So I would say, of course, it should have more surface area.
Surface area to volume ratio, when you are going to check, then obviously there is a difference.
Surface area to volume.
Volume ratio, and overall, if you will check it, you will get to know. Small things, more surface area. The exchange is comparatively easy.
Okay.
I'll teach you after the class.
Okay.
Right?
Okay. I'll be here.
I'll teach
I'm
him.
learning, guys. I'm learning.
Okay? So if a cell is small in size, right, its surface area is more, and in comparison to that, its volume is less. Okay, so easily the exchange with the surrounding is possible. If something has to come inside, if something has to go, you know that. Okay, so when the cell will increase in its size, cytoplasmic growth is there. And what is going to happen here? What is going to happen here? The volume will increase and the surface area will be less. And that surface area is not sufficient to draw the material from the surrounding. Okay, so cell has to divide. That's why division is there. So now our main or the actual topic is cell cycle. And when we talk about the cell cycle, it's a sequential, right? It's a sequential process. In a sequence, the things will be. Sequence the things will be. So what is the meaning of being in a sequence? Like you know that cell growth will be there. That will continue throughout. Plus DNA replication will be there. But that will be in a particular phase only, right? It will not be for the complete time period, but it will be there for the limited period only. What is this? Yeah. You use my right profile. Okay. You use my right profile. You go there. So basically the point is, sir, you will see all of us together soon. Okay. So the point here is, in the case of cell cycle, it's a sequence of event. It's a sequential process. In a sequence, things will be there. So here the cell growth plus cell division will be there. So now what we need to focus, because of course it's a quick series. So interphase and M phase. That's what we read.
Metaphase.
Mitosis. Interphase or M phase. When you talk about this interphase, it is also known as the resting phase. It is also known as I phase. It is also known as invisible phase. It is also known as preparatory phase. Okay, there are different names for this particular phase. Now, when you talk about this M phase, it is the actual division phase. The cell division will actually take place here. Are you getting my point? The cell division will actually take place here, and it is the resting phase, I phase, invisible phase, and preparatory. But it is the most important phase, because a cell is going to spend more than 95% of its lifespan in the interphase. The reason is it has to prepare itself for the division. Now, I'll give you the example of your NEET preparation. You people spend 2 years, 3 years, sometimes 4 years for your NEET preparation. Why? Because you want to do well in that exam that is for 3 hours. So for 3 hours, so that you can perform well, you prepare for 2 years, 3 years, or 4 years. So you people can relate interphase like this, right? You are preparing. For other people, you are just doing nothing. You are attending the classes, you are just reading books, and you are solving the questions. But you know that you people are under immense pressure, isn't it? Isn't it? Volume increase करने के लिए बोल रहे हैं? दिक्कत है क्या कुछ? Okay, right? Getting it? Okay, done. So basically the point is, like, you people are preparing, and when you people prepare, you are under immense pressure because you have to do well in your exam. Okay, so same is the case here. In the interphase, the cell has to prepare, right? The cell has to make sure that in the division phase, there should be no error. Otherwise, mutations can occur, right? If mutations will be there, you know that disorders can occur, right? The chromosomal, I am not a disorder. Okay? So it is like that. Okay, so interphase or M phase. Now there is a line in the NCERT which states that interphase is the phase. I'm picking up all the PYQs here. Interphase is the phase in between two successive M phase. Interphase is the phase between two successive M phase. Two successive M phase means, two successive means one after another. Two successive means one after another. So basically here the point is, you have M phase, you have I phase, you have M phase. It's like that. So I phase is present in between two successive M phase. Now, when you talk about this I phase, we have G1 phase, we have S, we have G2. G1 stands for gap 1 phase. It stands for growth 1 phase. And because it is just before this S phase, in S, the DNA synthesis is there, right? So it is just before this S phase. So we also call it as pre-DNA synthesis phase. We also call it as pre-DNA synthesis phase. Got it? And here, this S, it is the synthesis phase. It is the only phase where the DNA replication will be there. We'll come to this. Now, what about G2? Again, it is the gap 2 phase. Now I am getting distracted. You leave. Growth 2 phase. Post DNA synthesis phase. Post DNA synthesis phase. Now, one more question, and that is a PYQ. See, it is NCERT's line that interphase is the phase between two successive M phase. Right? It is the NCERT's line that interphase is the phase between two successive M phase. So G1 is after M phase. G1 is
After M.
G1 is
After M.
so G1 will be
After M, please.
post mane after mitotic phase. Post mitotic phase it is. Post mitotic phase it is. Is that clear? Post mitotic phase it is. Is that clear? Now, when you talk about the G2, it is the pre-mitotic phase. So it is a PYQ. Many times students make the mistake. Remember this line and remember this part. M phase, I phase, M phase. You'll get the answer of your question. Okay, you'll get the answer of your question. And moreover, there is one more PYQ from this part only that, what about that?
This synthesis.
No. From that, so many questions are there. Yes. So basically, G1 phase, it is present in the interval in between the cell division. It is present in that interval. It is present in between M phase and synthesis phase, G1 phase. Right? So in between cell division and DNA replication, we have G1 phase. That's a PYQ. In between cell division and DNA replication, we have the G1 phase. That is also a PYQ. Okay? That is also a PYQ. Done? Sure? Okay. So now let's move to the next part. So G1 phase, what is going to happen here in G1 phase? Normal. Protein synthesis, RNA synthesis. And remember, whatever is required for S phase, everything will get synthesized here in G1 phase. It's like we need something in G2 phase, that will be done here in G1 and S. It's like that. Right? It is like that. Oh, देख रहे हो तारीफ है आपकी. Good, better and best explanation होता है सर जी आपका. सर बायोलॉजी पढ़ो सर से. Explanation समझ में आएगा.
I'm learning. I got 2 MCQs.
Actually, you'll be able to answer them. So now
G1 is between M and S.
So what is going to happen in G1 phase? It's a gap 1 or growth. So basically, when the cell is in interphase, in the resting state, so what is going to happen?
Carry on.
Now you got bored. Okay. So when the cell is in the interphase, okay, so what it has to do, it basically the cytoplasmic growth will be there, and that will be throughout. That will be throughout the cell cycle. DNA replication will be there only in the S phase, but the cytoplasmic growth will be throughout the cell cycle. That's what you need to keep in your mind. Right, bache? That's what you need to keep in your mind. And Aisha, right, for today's lecture only, we are going to cover these topics. Otherwise, we have other topics also. If you remember, in 4 lectures, remember, top 50 concepts, 4 lectures. So that's how things are. Okay? Right? That's how things are. So keep it in your mind. Okay? So when it is the G1 phase, so as I said, here the RNA synthesis, all that essential proteins, that nucleotides, nucleosides, ATP synthesis, enzymes, etc., they all will get synthesized here. They all will get synthesized here. But the one thing that you people need to keep in your mind is, right, that here in the this G1 phase, remember this thing, please keep it in your mind, right? Or overall in the interphase, whenever you talk about the genetic material here, my dear students, it is not in the compact form like this. The chromosomes are not in this form, or the chromosomes are not in this form. They are not that compact. Okay, they are not that compact. Okay, they will be in the thread-like form, the loose threads, the entangled one. Okay, so we call it as chromatin. So it is there in the thread-like form. Okay, so overall, when you talk about the genetic material here, right, it is in the thread-like form, and there will be single arms, right? It will be having one arm only. So they all are, right? They all are tangled. It is not like you can look at it individually. No, not like that. Right? Not like that. Individually, they are not present, students. They are entangled. But the point is, whenever you will talk about the chromatin here, it will be having single arm. It will be having single arm. Right? So it means it is monad. Single arm is there. Are you getting my point? Single arm is there. Are you getting my point? So as I said, in the G1 phase, RNA synthesis, protein synthesis, all the essential enzymes, ATP, ADP, whatever is required for the next phase, they'll be there. And then comes S phase. Then comes S phase, that is synthesis phase. So it is the phase in which what will happen? DNA duplication. Right? These are the keywords that I'm writing here. DNA duplication will be there, plus even the centriole duplication will be there. What will be there? Centriole duplication will be there. So what is centriole? Anybody here in the class? What is centriole? Anybody here in the class? Anybody here in the class? What is centriole? Yes. What is centriole? Yes, please. Anybody? So centriole, you know that it's a non-membrane bound organelle. What's that? It's a non-membrane bound organelle. And where you people are going to see this? In the animal cell. Okay, let me change the color as well. So you people are going to see it in the animal cell. You people are going to see it in the animal cell. Got it? You people are going to see it in the animal cell. Okay. Okay. So DNA duplication and centriole duplication will be here. And that is also NTA's favorite question. Right? You can write it like this. It is NTA's favorite question. So people, 3 to 4 questions are expected, right? 3 to 4 questions are actually expected from this particular chapter, this particular topic overall. Done? Okay. Chali. Let's go. Let's discuss it further. So, as I said, DNA duplication will be there. Just for an example, if the initial amount of DNA, if it is 2C, initial amount, initial quantity, initial concentration, right? Just let's say 100 picogram it is. So, in S phase, it will become 4C, 100 will become 200. That's what I'm trying to say. 100 will become 200, right, bache? So initial amount of DNA will be 2C, then it will become 4C, but the number of chromosome will remain same. But the number of chromosome will remain same. Number of chromosome will remain same. That's what we have. That's what we have. So I hope this part is clear, right? I hope this part is clear. Right? The number of chromosome will remain same, but the DNA amount will get double. Right? The DNA amount will get double. Okay? Is that clear? I hope this part is clear. So if it is not clear, let me give you a basic idea here. It's very simple, students. Let's suppose this is a cell. Imagine this cell is having 4 chromosomes. I usually take this example. 1, 2, 3, and 4. How many chromosomes are there? 4. No doubt when a cell is not dividing. No doubt when a cell, it is not dividing. It is normally, when a cell, it is not dividing, no, the genetic material is in the thread form. It is entangled. It is in the thread form. Indeed. But now just to explain you this, I'm using this example. So let's suppose this cell is having 1, 2, 3, 4 chromosome. So in the S phase, how is it possible that 2C will become 4C, but the number of chromosome will remain same? It's simple. It's simple, bache. Right? Ultimately, if you talk about a chromosome, what does it contain? DNA. Yeah, DNA is there. Proteins are there. So basically the DNA coil over the proteins, then they get compacted, right? Then they get condensed, then they become compact, then they get condensed. So here in the S phase also, bache, the DNA content will get doubled. So that's why this monad will become diade. This monad will become diade. So 1, 2, 3, 4, right? This monad will become diade, right? Right. So initially, if the chromatin threads are like this, then later on they'll become this. Later on they will become this. So monad will become diade. So DNA amount will get doubled. Here, the DNA amount is doubled, but the number of chromosome they are same. That's the point here. Okay, that's the point here. That is in the S phase. And histone proteins, right? So this part clear, right? This part clear about the S phase. Then comes the G2 phase. In the G2 phase also RNA and protein synthesis is there. In the G2 phase also RNA and protein synthesis is there. But the point is, right? But the point is, students, that in the G2 phase, like RNA synthesis, protein synthesis is there. Plus one thing that you can specifically mention, that tubulin protein synthesis will also occur here, because the rest everything is same. So what's the point here? Tubulin protein synthesis will also take place here. Okay, tubulin protein synthesis will also take place here. Okay, so this tubulin, you know that it will polymerize, it will form microtubules. That microtubules are very important for the spindle apparatus. That is also a PYQ. Is it done and dusted? Is it mentioned there in the chat? Yes? Is it mentioned there in the chat? Is it mentioned there in the chat? Yes, please. Yes, please. Yes, please. Sure. Sure. Are you sure? So in the G2 phase, there will be the tubulin protein synthesis. The tubulin protein will get synthesized here. Okay, the tubulin protein will get synthesized here. Rest RNA synthesis, protein synthesis, it will be there in this particular phase. Now, students, when you talk about this part, G1 phase, when it comes to the duration, it is the most variable phase. Its duration is not at all fixed, right? It can change. It depends upon the duration timing, of course. So let's say if a cell is dividing very quickly, the G1 phase will not be long lasting. If it is taking time, G1 phase will take time. So it is like that. So DNA replication is only there in S phase. Centriole duplication is also there in S phase. So that is NTA's favorite question. That is NTA's favorite question. Okay. Now let's come to the actual topic, that is the M phase, and M here means mitosis. M here means mitosis. So how many of you hate mitosis? How many of you hate mitosis and how many of you love mitosis? Today, the session speed is going to be like this. I have to be quick, students, because we have to complete the many important questions today. Okay, we have to do it. We have to do it. Yes? Done? So how many of you hate mitosis? Bache, from this three, easily, bache, 100 plus. Mitosis is the love of my life. No hate. I'll marry mitosis. Matlab, that is not possible. That is technically not possible. Okay, so if you love mitosis, then I want to see some fire emojis in the chat section. Bache, Shiva Prasad, I'll come to that also. If you love mitosis, so I want that some fire emoticons here in the chat section. Quick. I want some fire emoticons here in the chat section. Quick. Quick. Yes. Some more. And guys, if you're new to our channel, do not forget to hit the subscribe button. Okay, so mitosis. We know that it is the actual division phase, and this phase is known as equational. This division is known as equational division. People, is that okay if I just explain you the concept rather than highlighting the NCERT, because I have done it so many times? And let me explain this thing. Let me mention this thing to you people again, that people listen to me. We will finish this important topic series, then in the NEET endgame, as we promise, all the chapters will be completed in 2 to 3 hours. We will do it. And already in Game of NEET 2.0, we have completed many chapters. So whatever is left in Game of NEET 2.0 and in the NEET endgame, that will be covered there in the, that will be covered further, right? That will be covered further. So it's not like that we are going to end some series. Yeah, we are not going to continue that. We will. Ultimately, we have to complete your syllabus and that will be done. Don't worry about that. Okay, so it is the equational division, means parent cell will divide into 2 daughter cells. So parent cell is also diploid, daughter cells are also diploid. Parent cell, it will divide into daughter cells. So parent cell and the daughter cells, they will have the same chromosome number, right? Parent cell and the daughter cells, they'll have the same chromosome number, right, bache? So that is why it is the equational division. So what we have to remember here, that when you talk about the mitosis or the meiosis, what do we have here in mitosis and meiosis? What do we have here in mitosis and meiosis? So we have karyokinesis and we have cytokinesis. We have karyokinesis and we have cytokinesis. We have karyokinesis and cyto. Karyon mane nucleus. Kinesis mane division. Karyon mane nucleus. Kinesis mane division. Cytokinesis, cytoplasm division here. Right? Cytokinesis, cytoplasm division here. Right, it is the cytoplasm division. So karyokinesis is the nuclear division and cytoplasmic division. So karyokinesis will have PMAT. What will you see here? You will see PMAT here. You will see PMAT here. What will you see? PMAT here. Okay, so prophase, metaphase, anaphase, telophase. And your Wasim sir was saying M phase is metaphase. So kindly do not learn biology from him, but he's very good in chemistry. I hope he's not listening, and I hope he's not watching this lecture. So he's very good. He's a very good chemistry teacher, right? And of course, his voice is also amazing, right? That is why students, they are like, oh my God, what a voice sir is having. That's true, by the way. But yeah, he's a worst teacher of biology, right? I can still teach chemistry. I can still teach physics, but he cannot. He cannot. That's true. So that's PMAT. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Now directly come to the PYQ section here. So I have already explained you what G1 is. I have already explained you what S is. I have already explained you what G2 is. Okay. Now there is a point, right? There is one more thing that is known as G0. There is one more phase that is known as G0 phase. So G0 phase is cell arrest phase. It is quiescent stage. It is cell arrest phase and it is quiescent stage. It is cell arrest phase and it is quiescent stage. What is this? G0 phase. So let's say, because all the cells, they do not divide. For an example, you talk about the nerve cell, you talk about the heart cell, they'll only form during the embryonic development, etc., etc. They'll not divide further. So what will they do? So when they are in their G1 phase, if they will not get a go ahead signal, right? Because here in the G1 phase, there are go ahead signal. Like in some organisms, it is known as the start point. In some organisms, it is known as the restriction point, right? So when the cell will get a go ahead signal, that cell will cross that particular phase and then it will enter in the S phase. And once a cell enter in the S phase, then for sure it will complete the cell cycle. For sure it will complete the cell cycle. Right, bache? Right. But if it will not get the go ahead signal, they will leave G1, they will enter in the G0 phase. So this G0 phase, it can be temporary, it can be permanent. Temporary means a cell is there in the G0 phase for the some time. A cell is there in the G0 phase for the some time. Matlab, that is a temporary G0 phase. Cell is there in that particular phase for some time only, right, bache? And when there will be the signal for the division, it will further enter in G1 and then will continue it. And it can be permanent, means cells, they just divide only during the embryonic growth. Otherwise, they do not. So it is like that. Okay. It is like that, and that is what you need to keep in your mind. Okay. That is what you need to keep in your mind. Any doubt? Any doubt? Yes. Any doubt? Yes. So temporary is temporary, जैसे for an example, Arjun, if you talk about the humans, right? These are general things. In the case of human, you know that within 24 hours, the cell will divide once, and that too for 1 hour. Remaining is the interphase. Okay, remaining is the interphase. It's not like that all the cells dividing every time. It depends upon the body need. Let's say for an example, right? Let's say for an example, if you will get a cut here, right? The healing has to be done here. The healing has to be done here. The new cell growth is required. So the cells present here, what will they do? The cells present here, what will they do? They'll start dividing because they need to heal, right? They need to heal. So they will enter in the cell division phase. It's like that. So some cells in temporarily, some cells, they are in the temporary G0 phase, some cell in the permanent G0 phase. Okay, just say heart cell, nerve cell, permanent G0 phase. They will not divide further. Okay, they will not divide further. Okay, so that's what you need to keep in your mind. Then checkpoints not given in NCERT, but again important, like from G1 to S, there is a restriction point, right? So cyclins, they are the proteins, and CDKs, they are the cyclin dependent kinases. They are the cyclin dependent kinases. They are the cyclin dependent kinases. They are the enzymes. So periodically they make and break, and that's how the cell will further proceed. So G1 checkpoint is there, G2 checkpoint is there, right? And because if let's say proper DNA synthesis is there, everything is proper, then obviously G2 checkpoint will be like everything is fine, then you enter in the M phase. So G2 to M transition, G2 to M transition is also having a checkpoint. So there are checkpoints which will make sure, that the cell growth is proper, the cell division is proper, the error should not be there. The errors should not be there. Right? Right? Okay. Chaliye. So now, as I said, let's come to the topic directly. And this is the NCERT diagram. Do not forget about it. And that's your homework. People, do solve this part. Okay. Do solve this part. This is your homework. These tables are your homework. And multiple times I have discussed that in my one-shot sessions. Okay. So, as I said, directly come to the point. Mitosis, equational division. So, haploid cells, do they show mitosis? Do they show mitosis, Shweta? If you will follow the session regularly, if you will complete this particular session, yes, you can boost your marks, right? You can boost your 100 marks as well. But please pay attention. Please answer my question. Jwalit will come to this oogenesis part as well. Allow me some time. In meiosis, we will discuss it. In meiosis, we will discuss it. Yes, Bache Himansh, thank you so much for this. So, what are we talking? What are we talking? We are talking about the prophase. Basically, we are talking about the division phase. And as I said, when you talk about the division, karyokinesis, nuclear division, you have PMAT. What do you have there? You have PMAT. What do you have there? You have PMAT, right? You have PMAT, that is prophase. That is prophase, right? First is the prophase. Now, I asked you one question that when I talk about mitosis, so even the haploid cells, my dear students, even the diploid cell, both type of cells can show mitosis. Both type of cells can show mitosis, because you know that in the case of plants, right? In the case of plants, algae, the main thallus is haploid. In algae, main thallus is haploid. So definitely mitosis is there. Definitely mitosis is there. Diploid cells, they also show that. So mitosis can be there in haploid cell as well as in the diploid cell. Keep it in your mind. Okay. Just keep it in your mind. Okay. So now what do we have in the prophase? See, this process is very simple. This process is very simple. If I have to repeat it, let's say prophase. Let's say prophase, metaphase, anaphase, telophase. In this particular slide only, I'll explain everything, right? But as I said, we'll cover the most important things here. Let's say there is a cell where 2n is equals to 4. Let's say there is a cell where 2n is equals to 4, where 2n is equals to 4. So what is the point that we have? We are talking about the PMAT. So prophase, metaphase, anaphase, and the telophase. So in the prophase, basically the nuclear membrane, the chromatin will start the condensation. Okay, the chromatin will start the condensation. Normally you can see this. You can see this. The genetic material is entangled there, right? This is tangled there, right? This is in the form of loose thread, but in the prophase, its compaction will start, right? It will become compact. It will start condensation. It will start condensation. It will become compact. Now, in the animal cell, you know that we have the centriole here, or I would say centrosome. Do you know what is centrosome, people? Yes. Do you know what is centrosome? Anybody here in the class? Do you know what is centrosome? But centrosome means, see, centrosome means when two centrioles, right? What is the centrosome? When two centrioles perpendicular to each other, plus they are covered with pericentriolar material. Two centrioles which are perpendicular to each other, plus they are covered with pericentriolar material, right? Pericentriolar material. So what do we call it? What do we call it? We call it as centrosome. It is also known as cell center. It is also known as cell sphere. It is also known as diplosome. Diplosome. Okay. So it is there for the formation of spindle apparatus. It is there for the formation of spindle apparatus. Is that clear? It is there for the formation of spindle apparatus. Are you getting my point? Are you getting my point? So of course, in the prophase, these centrioles, right? These centrioles, basically centriole will provide the orientation to the spindle apparatus, and this pericentriolar material, it helps in the polymerization of tubulin protein. It forms the microtubules. it formed the microtubule. Is that clear? So basically from this part you will see small tubules, they will start arising. Small tubules, they will start arising. Yes, they'll start arising. These centrioles will move apart, right? So by the end of prophase, you people will see that these centrioles, they'll reach to the opposite pole and they'll start forming these spindle fibers. So remember one thing, when it is the animal cell, right bache? So when it is the animal cell, here you will see some astral rays will also arise, right? Some astral rays will also arise. Some astral rays will also arise, right? Are you getting my point? Astral rays will also arise. Okay, and along with that, these are the fibers. Some are continuous, some are discontinuous. Okay, so by the end of the prophase, you will see the nuclear membrane will be totally disintegrated, right? The compaction of chromosomes is the compaction of chromosome will be there, right? They'll be there in the cytoplasm. They'll be there in the cytoplasm. All the nuclear, all the organelles, right? They will also disappear. Okay, and basically you'll see that's how the things are. That's how the things are. Basically, nuclear membrane is disintegrated. So the genetic material will be there in the cytoplasm. Spindle apparatus formation is already done. Spindle apparatus formation is also there, right, by the end of this phase. Okay, so that's what you have to focus. That's what you have to focus. So before discussing this part further, remember, you are talking about the chromosome here and they have two arms, right? They are in diade state. They are in diade state. They are in diade state. So this central part is the centromere. This central part is the centromere. What's that? It is centromere. So this centromere is what? We call it as primary constriction. We call it as primary constriction. We call it as primary constriction. And here what do you have? These are two sister chromatids. That's how the things are. These are two sister chromatids. That's how the things are. Okay, and you know that this centromere from both the sides, it is covered with what? It is covered with kinetochore. It is covered with what? It is covered with kinetochore, right? It is covered with what? It is covered with kinetochore. So that's how it is. And remember, the spindle fibers, they attach to this disc-shaped protein, not directly to the centromere ever, right? It is attached to that disc-shaped protein, but not directly to the centromere. Yes, done. So that's how things are. So in the prophase, this is the first stage, right? So kindly keep it in your mind. Kindly keep it in your mind that chromosomal material, they will condense to form compact mitotic chromosome and they have two chromatids attached together at the centromere. Two chromatids attached together at the centromere. Two chromatids attached together at the centromere. Understood? Understood, right? Then next is centrosome. Now you know what is centrosome. Now you know the difference in centrosome and the cell center. So centrosome, which had undergone duplication during interphase, they begins to move towards the opposite poles of the cell. They begins to move towards the opposite pole of the cell. So each centrosome will radiate out some microtubules called ester. So bache, when these centrosomes are there, they'll form these type of microtubules. We also call them as astral rays. So collectively, this structure is the ester. Ester means star-like. And where do you see this? You see it in the animal cell. Why? Because animal cell is having centriole, but plant cell is not having that. Plant cell is not having that. Okay. Animal cell is having the centriole. Okay. So two esters together with spindle fiber, they are known as mitotic apparatus. This question can come in paper. This question can come in paper for sure. This question can come in paper for sure. Understood? Sure? Are you sure? Yes, please. Any doubt? Any doubt? Okay, let's. And as I said, by the end of prophase, right? No Golgi complex, no endoplasmic reticulum, no nucleolus, no nuclear envelope will be there by the end. Now in the metaphase, what's going to happen? Remember this diagram, that is also a PYQ. This is not the metaphase, this is the transition to metaphase. This is the transition to metaphase. So when you people talk about the metaphase, in the case of metaphase, one thing is One thing that you need to keep in your mind is, see, let's suppose that's the point. Right? See, these chromosome in the metaphase, they'll align itself at the equator. Equator mane cell center. What is the meaning of equator? Equator means cell center. So all the chromosome will align itself at the equator. Right, bache? Oh, at the equator. It is also known as the metaphasic plate. No doubt in my diagram, it is not appearing like that, but that's how it is. Okay, that is how it is. So at the center, all the chromosome will align itself. So metaphase is the stage where we can study the shape, the size, the number of chromosome plus their morphology. That's it. Now in the anaphase, right, it is next to the metaphase. There is the splitting. There is the splitting. There is the splitting of centromere. Centromere will split, sister chromatids will get separate. But what's the point here? Their arms will trail behind. Their arms will trail behind, right? Their arms will trail behind. Okay, their arms will trail behind. Their arms, yes please, will trail behind. So in the anaphase of mitosis, what will be there? There will be the splitting of centromere, separation of sister chromatids, and arms will trail behind. Okay, that's how it is. That's what you need to keep in your mind. And in the telophase, which is again opposite to the prophase, again the nuclear membrane will form, right? Again the nuclear membrane will form. Again the chromosome will get decondensed. Again the organelles will appear. That is what telophase is. And after that, there will be the cytokinesis. So these are the main points related to the division, right? This is the metaphase stage. This is the anaphase stage. This is the telophase stage. So as I said, in the telophase, it is the reverse of prophase. So chromosome will cluster at opposite spindle poles and their identity is lost as discrete elements. Means we will not be able to see them as individual chromosomes, right? They will again be tangled. Okay, so nuclear envelope will form around the chromosome again at each pole that will carry two daughter cells. So nucleolus, Golgi complex, and endoplasmic reform, endoplasmic reticulum reform will be there, right? So nucleolus, Golgi complex, and endoplasmic reticulum will reform. Okay, okay. Done. So when there is the cytokinesis, if it is an animal cell, the contractile ring will form. Basically, there is a formation of cleavage furrow, bache. There is a formation of cleavage furrow, and it is the microfilament. It is the microfilament, yeah, actin filaments that help, right? That help there. And in the plant cell, it is simply, right, the cell plate that will form in between. In the plant cell, there will be cell plate, students, that will form in between, and Golgi apparatus will form, Golgi vesicles, will form some vesicles that will fuse in the center. They'll form the cell plate. So from the center, it will move towards the periphery. It will go towards the periphery. So centrifugally, it will form, right, bache? So this is what cell plate is. Got it? This is what cell plate is. Got it? This is what cell plate is. Got it? So in plants, there is the cell plate. Okay, chaliye. Now come to the meiosis. Meiosis is the reductional division. To the point we have to discuss, then we'll shift to the another topics of human physiology. Okay, we'll shift to the another topics of human physiology. Let's say if in any of the topic, if you guys are weak, if you think that, ma'am, I don't know this topic in detail, because here we have to discuss the things very quickly. Are you getting my point? We have to discuss the things very quickly. So if there is any topic which is important, plus you are not able to understand it here in this lecture, maybe you are doing it for the first time, so you can watch our regular lectures as well. No worry about that. Right? No worry about that. Okay? Okay? Done? Done? Take? Okay, chaliye. So next is the meiosis. Again, the reductional division. So remember one thing. We have interphase, we have M phase. Right? Here also the story is same, because this M phase is the actual division phase. So interphase is the resting stage, I told you already. And as I said, G1S and G2 is there. As I said, G1S and G2 is there. As I said, G1S and G2 is there. Is that clear? G1S and G2 is there. Is that clear? right? Then comes the M phase. So in the case of M phase, right, you know that what do we discuss basically? We talk about the meiosis. We talk about the meiosis 1 and then we discuss the meiosis 2. But what you have to remember here, right? That's a direct PYQ. What you have to remember here, that here in meiosis, there will be 2 rounds. There will be 2 rounds of nuclear division followed by cytoplasmic division, followed by cytoplasmic division. 2 rounds of nuclear division followed by cytoplasmic division, and 1 round of DNA replication only. 1 round of DNA replication only. Is that clear? Is that clear? So there will be 2 rounds of nuclear division followed by cytoplasmic division and 1 round of DNA replication only. Is that clear? So what is going to happen here? Yes, G1 is there, S is there, G2 is there. Then what will be there? Meiosis 1, meiosis 2. So one trick is same here, PMAT. But here it will be prophase 1, metaphase 1, anaphase 1, telophase 1. Here it will be prophase 2, metaphase 2, anaphase 2 and telophase 2. That point is same. That point is same. So how do we need to remember? I'm writing it here. Let's say for an example, you have meiosis. You have meiosis 1. Okay. So prophase 1, metaphase 1, anaphase 1, telophase 1. Then meiosis 2, right? Prophase 2, metaphase 2, anaphase 2, telophase 2. Prophase 2, metaphase 2, anaphase 2, telophase 2. Now point here is that here it is the prophase 1 which is the longest phase here, right? So here what will you see? You will see LZPDD. You will see LZPDD. This studio is as per Wasim sir's height now, so now I have to do this. Take, so it is LZPDD. It is LZPDD. Prophase 1, LZPDD. Little Zara, please do not dance. Little Zara, please do not dance. Leptotene, zygotene, pachytene, diplotin, dikinases. Leptotene, zygotene, pachytene, diplotin, dikinases. Leptotene, zygotene, pachytene, diplotin, dikinases. Okay. Leptotene, zygotene, pachytene, diplotin, dikinases. Got it? Got it? And now here in between. Now here in between telophase and prophase 1, this gap, sometimes there is a gap. It is interkinesis. It is interkinesis. What's that? It is interkinesis. What's that? It is interkinesis. Got it? Yes. It is interkinesis. Okay, got it? So the DNA replication will take place here only, but the nuclear division and the cytoplasmic division will occur. Nuclear division and the cytoplasmic division will occur. So 2 rounds of nuclear division, 2 rounds of cytoplasmic division, but there will be only 1 round. There will be only 1 round. 1 round of what? There will be only 1 round of this DNA replication. Understood? Now, the NTA's favorite question. Remember, if it is a prophase, definitely the DNA has to, the genetic material has to compact, the chromosome has to condense, the nuclear membrane has to disintegrate, the centrosomes, they need to move apart, that kind, spindle apparatus need to form. These are the things that need to happen for sure. But because it is the reductional division, it is going to reduce the chromosome number to half. It is going to reduce the chromosome number to half. It will reduce the chromosome number to half. So you have 1 cell diploid. So it will divide after meiosis 1 into 2 haploid cells. Then there will be meiosis 2 and you will get 4 daughter cells. So after meiosis 1, you will get 2 haploid daughter cells. After meiosis 1, you will get 2 haploid daughter cells. After meiosis 1, you will get 2 haploid daughter cells. And after meiosis 2, you will get 4 haploid daughter cells. Right? You will get 4 haploid daughter cells. So meiosis 1 and meiosis 2 is there. Meiosis 1 and meiosis 2 is there. Meiosis 1 and meiosis 2 is there. So in meiosis 2, these parent, they will become the parent cell. They'll give the daughter cells having same number of chromosomes just like parent cell. That's why meiosis 2 is compared with, that's why meiosis 2 is compared with, it is compared with what? That is why meiosis 2 is compared with, it is compared with mitosis. Okay? So here the chromosome number needs to reduce. So let's suppose there is a cell where 2n is equals to 8. So what are the things that we have to study there? First one will be leptotene. That's the direct question. So I am highlighting the the repeated PYQs from this part right now. Okay. So as I said, right, as I said that it is the leptotene. So of course, what is going to happen here? The chromosome will become gradually visible under the light microscope. This line is important. Under the light microscope, they will be visible. Under the light microscope, they will be visible. Okay. After that, the compaction of chromosome will continue throughout the leptotene. It will continue throughout the prophase rather, but throughout the leptotene, this will continue. This will continue. Now, you know that in some stages, we also say that, we also study that leptotene is also known as the bouquet stage, right? Bouquet stage. Why is it so? Nucleus, there is a chromosome plate, right? Like here, all the chromosomes will attach. It appears like a bouquet. In the insects, and there are some other cells where you will see the bouquet stage. So leptotene is sometimes also referred to as the bouquet stage. Leptotene is sometimes also referred to as the bouquet stage. Is that clear? Leptotene is also referred to as the bouquet stage. Is that clear? Then there is the zygotene. Then there is the zygotene, right? Then there is the zygotene. So here, zygotene mein, what is going to happen? The synapses. The synapses. What is going to happen? Synapses. In the zygotene, synapses will be there. In the zygotene, synapses will be there. What is the meaning of synapses? It means there will be the pairing of chromosome. There will be the pairing of chromosome. What will be there? There will be the pairing of chromosome. So pairing of homologous pair of chromosome. Just say for an example, if I just said that 2n is equals to 8. So you know that 4 chromosomes will be maternal, 4 chromosome will be paternal, 4 chromosome will be maternal, 4 chromosome will be paternal. So they will get paired. Let's say like this. Right now, I'm not telling you kinetochore is this is this, this is that. No, I'm simply showing you the pairing. I'm simply showing you the pairing. So they'll come closer. They'll come closer. They'll pair. They have to come closer, maternal and paternal, homologous chromosome. So do you understand homologous chromosome? Having same centromere position, having same shape, same size, same genes. Genes bhi same hai. They have the same genes. Alleles can vary. Alternative form can vary. Like for an example, this is the gene locus. Means the position of a gene on this particular chromosome. This is the duplicated arm only. So let's suppose in a layman language, if I have to explain, if here you have the gene for the height, here also you will have the gene for the height. Here also, if you have the gene for the height, here also, right? Right. Here also there will be the gene for the height. Here also there will be the gene for the height. Okay, bache? Okay. So homologous pair of chromosome, they are the one having same shape, same size, same centromere position. Right. The genes are also same, but the allele can vary. Right. Allele can vary. So pairing will be done. The pairing, the chromosomes are coming closer, right? There should be something which can hold them together. Just for an example, this is one chromosome, this is another. When they're maternal chromosome, paternal chromosome. So when they're coming closer, they need to be together, right? They need to be together, right? They need to be together, right? They need to be together. They need to be paired. So what's going to happen in between? There will be the synaptonemal complex. There will be the synaptonemal complex. There will be the synaptonemal complex. What will be there? There will be the synaptonemal complex that is going to hold them together, having RNA, protein, and that's how it is. They'll be paired. They'll be paired. So we also use the word. Hello. That's it. Okay. So this is what you need to know. So in the zygotene, do we have the synapses? Yes. Do we have the synapses? Yes. Right. Do we have the synaptonemal complex? Yes. Yes, bache. Right. Right. Is that clear? Is that clear? So synaptonemal complex will also be there. synaptonemal complex will also be there. Take synaptonemal complex will also be there. Will also be there. Is that clear? Is that clear? Is that clear? So, acha, that is the different thing now. So, firstly, pairing will be done. So, I am just showing you this, right? So, in the zygotine, the pairing will be done. What will be done in the zygotine people? What will be done in the zygotine people? The pairing will be done. Okay? The pairing will be done. Okay, so this is what synapses is. So, such paired chromosomes, they are called homologous chromosome, right? So electron micrographs will indicate, right, that here there is the formation of synaptonemal complex as well. There is the formation of synaptonemal complex as well. Is that clear? There is the formation of synaptonemal complex as well. Is that clear? Sure? Sure. Right? So synaptonemal complex will form, right? And you know that because compaction is already going on, right? Because the compaction is already going on. The chromosomes, they are condensing. These chromosomes, they are condensing also, right? They are becoming compact. Okay. So when you will look at it, it gives an appearance. It's a paired now. It's a paired thing. So they are paired. They are paired. Right, two homologous chromosomes, one is maternal, one is paternal. So, of course, when you talk about this paired one, when you talk about this particular part, right, when you talk about this thing, okay, remember, it looks like what? Bivalent. We call it as bivalent. Bi means two, because it is a pair. Because it is a pair. Because it is a pair. Okay, okay, bivalent. Or we also call it as tetrad. One student was asking this. We also call it as tetrad, right? We also call it as tetrad. Bivalent or tetrad. Bivalent or tetrad. Why tetrad, bache? Bivalent or tetrad is same. Because 4 sister chromatids are there, no? Because 4 sister chromatids are there, no? 4 sister chromatids. This is 4 sister chromatids are there. Are you getting my point? Are you getting my point? 4 sister chromatids are there. Okay? Okay, so it is bivalent or tetrad. It is bivalent or tetrad. It is bivalent or tetrad. Done? Done? Right? 4 sister chromatids. So remember, it is the homologous pair, right? So because these two are connected to the same centromere, so what do you call them? Sister chromatids. These two are also the sister chromatids, right? But when you talk about these two, they are non-sister chromatids of homologous pair of chromosome. They are non-sister chromatids of homologous pair of chromosome. Am I clear? They are non-sister chromatid of homologous pair of chromosome. Is that clear? So this pairing is there in zygotine, right bache? Right bache? And synaptonemal complex will be there. Okay, it is going to hold them together. We call them as bivalent or tetrad. So these keywords clear because question will come. So however, they are more clearly visible at the next stage. Matlab in the next stage, that is your LZPDD packaging. That is your LZPDD packaging. Anji Marish, that is your LZPDD. That is packaging. Okay, so packaging is the next stage, which is comparatively in comparison to leptidine and zygotine, its duration is more. Okay, so they are relatively short-lived compared to the next stage, that is packaging. Okay? So in the packaging, what will happen, bache? Recombination. We call it as crossing over. Okay. In the packaging, what is going to happen? The recombination will be there and we call it as crossing over. Just for an example, this. This is what we have. Yes, people. This is what we have. This is what we have. Yes. So what you have to remember here, see, crossing over means exchange of segment. It is also known as recombination. Crossing over means exchange of segments. It is also known as recombination, right? And which enzyme is required for here? Required here, it is recombinase. Which enzyme is required here? It is recombinase. So let's say, and always remember, that is again a P by Q, that whenever you talk about the crossing over, that exchange is in between the non-sister chromatids of homologous pair of chromosome. That is in between the non-sister chromatids of homologous pair of chromosome, right? Pair of chromosome. Non-sister chromatid of homologous pair of chromosome. That exchange is in between non-sister chromatids of homologous pair of chromosome. Non-sister chromatid of homologous pair of chromosome. So if at the place of non-sister, sister chromatids of homologous pair of chromosome is written, then it is incorrect. If non-sister chromatid of non-homologous pair of chromosome is written, then also it is incorrect. Like for an example, if you look at this part, the exchange is in between this one and this one, or this one and this one, but not in between this one and this one. Okay, these non-sister chromatids of homologous pair of chromosome will exchange their segment, but not the non-sister chromatid of non-homologous pair. This is the homologous pair, but when you compare this one with this one, this is the non-homologous pair. The exchange will not be there. I hope I'm clear here. Okay, so synaptal nemal complex done. Next phase will have the crossing over. So let's say if the crossing over has to be done at this point, so you will see before crossing over, before the exchange of segment, before crossing over, before the exchange of segment, people, before crossing over, before the exchange of segment, remember, let's say if at this point crossing over has to be done, so remember one thing, the recombination nodule will appear there. It is marking as it is the site at which the crossing over will occur. So up to this part, all clear? Are you sure? So recombination nodule will form. So after that, let's say, just imagine. So let's say if the exchange, now the exchange will occur. So the site at which the crossing over, like where it has to be done, right? The recombination nodule will form. It is before crossing over. It will mark the site, okay, this is the point from where the segment will get exchanged. So when they will get exchanged, what will you see? You see X-shaped chiasmata. What will you see? You'll see X-shaped chiasmata. What will you see? You will see X-shaped chiasmata. Understood? Okay, so X-shaped chiasmata will form there. Okay, so 4 chromatids of each bivalent chromosome, they'll become distinct and clearly appear as tetrad. They'll be very clear as tetrad in the pachytene, right? Next, what is going to happen, bache? As I said, recombination nodule will form, done. Crossing over, done. Okay, so after that, LZP done, then LZPD. LZP done. We have discussed leptotene, zygotine, pachytene. Now let's talk about the diplotene. So when you people talk about the diplotene, it is the dissolution of synaptonemal complex. So solve, check the paper, check last 5 years, right? Check the question papers of last 5 years. You'll get the questions from these part. At least 2 to 3 questions. One question is for sure from this LZPDD thing. Now, this is the favorite question of your examiner. Trust me, this is the favorite question of your examiner. Trust me on that. Okay, trust me on that. Okay, done. That's what you need to keep in your mind. Done. So as I said, the tendency of the combined homologous chromosome or bivalent to separate from each other except at the site. So basically, now imagine. I told you that imagine if these are two chromosomes, they are coming closer, synaptonemal complex has to hold it together. Synaptonemal complex has to hold it together. Now, if it will dissolve, right? If there is the dissolution of synaptonemal complex, don't you think they'll move apart? Don't you think they'll move apart? Don't you think they'll move apart? They will. They will. They will. But they will remain attached to each other only at one point, and with that you know the X-shaped chiasmata, which marks the site where the crossing over is done and dusted. Right? Where the crossing over is done and dusted. This is what they are marking. So X-shaped structure, X-shaped chiasmata. Okay, so because synaptonemal complex is dissolved, right, bache, the bivalent is trying to separate from each other, but it will remain attached to each other at the site of crossover. It will remain attached to each other at the site of crossover, right? The place where the crossing over has to be done, it will remain attached to that. Is that clear? So these X-shaped structures are called chiasmata. These X-shaped structures are called chiasmata, right? So in oocytes of some vertebrates, diplotin can last for months. Jaise ki even if you talk about the humans, in our case, you know that oogenesis, it starts in embryonic life. It starts in embryonic life, right? Matlab when the female, when she is in her fetal stage, her ovaries will start forming the gametes, okay, in her embryonic life, number one. It will start in her embryonic life, number one. Now, here in the embryonic life, meiosis will start. What will happen? Meiosis will start. What will happen? Meiosis will start. Yes or no? Yes or no? Right? But this meiosis will start, the prophase I will start, and it will be in L, Z, P and D. Matlab there will be an arrest in the diplotin stage. There will be an arrest in the diplotin stage. And we use the word dictate arrest for that. We use the word dictate arrest for that. We use the word dictate arrest for that. Is that clear? Is that clear? Sure? Sure? Done? Done? Okay. Okay. So next is the dikinesis, which is the final stage. It also marks the transition to metaphase. And here what's going to happen? The terminalization of chiasmata means, let's suppose if your chiasmata at this particular time, at this particular place, if this chiasmata has formed here, its terminalization will be done. It will reach to the ends. But from ends, it will remain attached. That's how it is. That's how it is. Okay. Now, students, this comparison you have to keep in your mind. When I started this topic, I told you, let's imagine there is a cell, right? There is a cell having 2n is equals to 8. So if the diploid number of chromosome is 8, so how many pairs will form here? 4 pairs. How many pairs will form here? 4 pairs. Okay? So right now I'm not getting into the technicalities. All I'm showing is that you know that finally in metaphase, in the metaphase I, yes. So in the metaphase I, in the metaphase I, in what? In the metaphase I, always remember there will be 2 metaphasic plates. There will be 2 metaphasic plate, right? 2 metaphasic plate, and the chromosome will align on the equator. The chromosome will align on the equator. 2 metaphasic plates are there. Actually, this bivalent is arranged here. Actually, this bivalent is arranged here. Actually, this bivalent is arranged here. So 2 metaphasic plates are there. 2 metaphasic plates are there. Bivalent will be arranged like this. Okay. Okay. So now you imagine this thing. If and yes, there is one word known as congression. Congression means these chromosomes, they are coming to the center and then they'll be arranged at the metaphasic plate. Right? What is the meaning of this congression word here? What is the meaning of this congression word here? The chromosome, they will move and they'll align itself at the equator. They'll form the metaphasic plate. They'll form the metaphasic plate. So bivalent will be arranged like this. Now, if there is the anaphase I, you tell me what's going to happen in anaphase I. You tell me what's going to happen in anaphase I. That is also PYQ, my dear people. That is also PYQ, my dear people. What's going to happen? What's going to happen? See, Aisha, first of all, bache, those who are not able to understand anything, it simply means that you don't know anything about the topic. If you want the lectures to be there from the basics and to be very slow, start from our Game of NEET series. Okay? Start from our Game of NEET series. Otherwise, it is of no use. We are talking about the top 50 concepts. We are assuming at least you know something. And even if you don't know, whatever topics I have picked, I'm starting it from the scratch only. But it is the biology. It is not physics and chemistry where I'll give you one numerical, where I'll explain one concept, I'll show you one numerical, and I'll say, okay, fine, that's how you have to solve it. Here we have the theory. We literally need to discuss the theory, and it takes more time. It takes more time. Okay? It takes more time. So kindly focus here. And if you cannot, then please do not waste your time. Done? Done? So I'm not here to, tell you, give you the feel, array, everything is easy, you'll do it. Array, yeah, theory it is. It will take time. Give some time. Okay, so if it is, this is the metaphase 1. So in metaphase 1, there will be 2 metaphasic plates. There will be basically bivalent is arranged there. Bivalent is arranged there. Now you tell me if it has to be the anaphase, what will be done? The separation of homologous pair of chromosome. Separation of bivalent is there. Separation of this homologous pair, you can say, or the bivalent. Or the bivalent. Right, separation of homologous pair or the bivalent. So here in anaphase 1, there is no splitting. In anaphase 1, there is no splitting. Right, in anaphase 1, there is no separation of sister chromatid. Rather, there is the separation of the bivalent. Rather, there is the separation of the bivalent. Baki telophase 1, it's the opposite of prophase only. You know that what is going to happen. You know that when the bivalent will separate, it will reach to the opposite pole. It will reach here, right? It will reach to the opposite pole. It will reach here. The basically decondensation will again occur. The again chromosome will lose their identity. Again they will be tangled, but not completely, because telophase 1 has immediately jumped to, after the cytokinesis, it has to jump to prophase 2. Okay, so when you people talk about the prophase, okay, you know that again the story is same. Prophase 2, metaphase 2, anaphase 2, telophase 2. Again the story is same. Prophase 2, metaphase 2, anaphase 2, telophase 2. The story is the same. The story is the same, right? But the point here is we compare it more with mitosis, because here there is no pairing and all. You know that you have n number of cells. They'll give you 2 daughter cells having n. So if let's say, if in the anaphase 1, there is the separation, if there is the separation of homologous pair of chromosome, don't you think this cell will have 4 chromosome and this cell will also have 4 chromosome? Don't you think this cell will have 4 chromosome and this cell will also have 4 chromosome? Don't you think so? Don't you think so? This cell will have 4 chromosome and this cell will also have 4 chromosome, of course, right? But you have to look at it like this. Just see, this cell will enter in the division phase. So what will be the amount of DNA here? 4C. Now they will divide into 2 daughter cell. What will be the amount of DNA here? 2C and 2C. Now after metaphase 1, you have 4 haploid cells. So what will be the amount? This is C, C and C. This is C, C and C. So that is something you need to remember. So this cell is having 4C, the DNA amount. Here it is 2C and 2C. Here 4 daughter cells are there. And then here also it is C, C, C and C. That's what you need to keep in your mind, right? That's what you need to keep in your mind. Okay. That is what you need to keep in your mind. Okay. Okay. Done. Sure. Sure. Pakka. So after telophase 1, of course, cytokinesis say, what will you see here? 4 chromosome here, 4 chromosome here. DNA amount is 2C and 2C. So just to separate that sister chromatids, because we have to make the DNA amount C also, right? So that's why we have the meiosis 2, right? Meiosis 2. So again in the prophase, same story, but here when it is the meiosis 2, right? Here when it is the meiosis 2, in the meiosis 2, what is going to happen, people? the chromosome will be aligned like this. Now, here you don't have 8 chromosomes, you will have 4. You will have 4, right? Because the chromosome number is reduced to half, my dear people. So they will be arranged at the equator like this. So now in the anaphase 2, according to you, what will happen? In the anaphase 2, again there is the separation of sister chromatids. How? Because there will be the splitting of centromere, right? Chromatids, because there is the splitting of centromere. There is the splitting of centromere, so there is the separation of sister chromatid. There is the splitting of centromere, and so there is the separation of sister chromatid. Okay, done? Done? Sure? Sure? Up to this part, all clear? So this is all about this division, right? I have highlighted all the important points. You can just watch this particular diagram as well. You'll get a better idea, and the same is in the NCERT. So you can just watch it, have a look, get an idea. If there is any doubt, now ask me. So basically, not just the 3 questions, we have covered 4 different topics, and yes, multiple questions can come from this part. Done? Done? Yes? Which is cytokinesis means cytoplasmic division. Interkinesis means in between, the gap that is there in between the 2 cell division. Just for an example, meiosis 1, meiosis 2, right? After telophase 1 and between prophase 2, there is a gap. That is interkinesis. That is interkinesis. That is interkinesis. Okay, chaliye. Now we will jump to the next topic, right? Excretory product and its elimination. We'll start with the urine formation. We'll cover every point from there. Okay, so I believe up to this part everything is clear. Now let's talk about this topic, that is urine formation. Okay, so what do you know about it? Do you know the nephron structure? Do you know the nephron structure? I'm not going to discuss that. Do you know the nephron structure? Arey beta, here, right? It's biology. You understand the concept, right? You already have the content available. Solve the questions directly, right? And the next lecture that we have on Sunday, in that particular lecture, we will revise the concepts with the help of PYQs only. Otherwise, there will be too much of explanation. So in the next class, that is on Sunday, I'm going to pick up the topics from class 12th, right? I'll definitely pick up the topics from class 12th. And in that class 12th topics, like I'll pick up 30 topics there. 30 topics means 60 MCQs. That's what we have to, right? That's what we have to do. Okay? Okay? Urine formation. So you understand. Do you understand the nephron structure? I believe you all know that. The nephron is the basic. Yes, it is the nephron. That is the basic structural and functional unit of what? Kidney. So that's how we draw it. And I believe you all understand this part. Done? And then you know that all the blood capillaries, they are going to cover it like this. Well, so let's discuss this. The topic name is urine formation, which will include 3 steps, right? Which is going to include what? 3 steps. So here we have the glomerular filtration, tubular reabsorption, and tubular secretion. These are the 3 steps: glomerular filtration, tubular reabsorption, tubular secretion. Glomerular filtration, tubular reabsorption, tubular secretion. These are the 3 steps that we have here. Okay, these are the 3 steps. So first step is definitely the glomerular filtration. So here you need to remember one structure, and that is what we have. Okay, that is what we have here. Just look at the diagram. Like if I have to explain it, it's simple. It's simple. It's like this. Right? This is the cup-shaped woman's capsule, and here the blood capillary, the tuft of blood capillary glomerulus is there. The tuft of blood capillary glomerulus is there. What do we have here? Tuft of blood capillary glomerulus is there. Okay. Okay, so this is the outer layer of Bowman's capsule. This is what? It is the outer layer of Bowman's capsule. This is the inner layer of Bowman's capsule, right, bache? And here you have the tuft of blood capillary, and that is glomerulus. That is glomerulus, right, bache? So see, blood capillary, renal artery, it will break. So this one is this is entry. This one is exit. This is something is coming. Ana, afferent atrial. Afferent atrial. What's that? That's afferent atrial. What's that? That is afferent atrial. This one is efferent atrial. So if you will look at the diameter, there is a difference. This one is wider, this one is narrower. And this is the tuft of capillary here. This is the tuft of capillary here. That is glomerulus. This is the tuft of capillary here. That is glomerulus. Right? This is the tuft of capillaries here. That is glomerulus. Is that clear? That is glomerulus. Is that clear? Right? Because the first step of urine formation will occur here. Why this difference in diameter is there? Why that difference in diameter is there? It's simple. It's very simple, right? Why that difference in diameter is there? Right? This difference in diameter will exert a pressure that we need for the filtration, right? That we need for the filtration. So here there will be the glomerular filtration, the ultrafiltration. So what you need to remember, the arrangement of the cell. See, if it is the tuft of capillary, the glomerulus. So if it is a capillary, what kind of cells will be there? Endothelial cells. Endothelial cells. And you will see that, let's suppose these are the cells. In them, there will be the pores present in between, the gaps, right? There are pores present in between. These are the gap. So what do we call them? Fenestrae. Am I right? We call them as fenestrae. And of course, they are lying on what? Basement membrane. We know that epithelial cells, they lie on basement membrane. Now, inner layer of Bowman's capsule. Inner layer of Bowman's capsule. This inner layer of Bowman capsule is also having specialized, or you can say that modified cell. And what do we call them? This inner layer. What do we call them, bache? They are the podocytes. They are the podocytes, right? They are the podocytes. So poda mane feet. Poda mane feet. Podocytes. Poda somewhere is associated with feet. So these cells are arranged in such an intricate manner, my dear people. These cells are arranged in such an intricate manner, my dear people, that there will be the slit pores in between. Okay? So we have the endothelial. These cells are also lying on the fenestrae. When you talk about the inner layer of Bowman's capsule, they are also having the special podocytes, bache, right? They are having the peat-like arrangement. So this layer, the cells here are also lying on the basement membrane. The cells here are also lying on the basement membrane, right? The cells here are also lying on the basement membrane. Is that clear? Is that clear? Is that clear? So basically, this endothelial having the fenestrae, these podocytes having the slit pores, and they both are lying on the basement membrane. So from here, the first step of the filtration will be done, and this is the ultrafiltration. So basically, the blood will get filtered out. So this diameter difference is helpful. It will exert a pressure here. Okay. So here, what will you get in between? Ultrafiltrate. Ultrafiltrate. So this ultrafiltrate is also known as glomerular filtrate. This ultrafiltrate is also known as glomerular filtrate. This ultrafiltrate is also known as glomerular filtrate, right? Done? Done, bache? Any doubt? Any doubt? Sure? Are you sure? So here, why these things will be there? There are certain pressures. There are certain pressures that will ensure these things. There are certain pressures, right? There are certain pressures. So now remember one thing, right? Here, because the ultrafiltrate will form. What will form here? The ultrafiltrate will form. What will form here? The ultrafiltrate will form. This ultrafiltrate, right? Yeah, glomerular filtrate. But it's like, it is not selectively passed here. Matlab, it is not like here the filtration is not something selective. Okay, this is what we have to pass. That is what we have to pass. No, it's random. Like pressure difference is there. Okay. Pressure difference is there, that is favoring the filtration. Indeed, that is favoring the filtration. And here, let's suppose in the blood, if there are certain plasma proteins which cannot cross this, only they will not pass. Only they will not pass. Otherwise, all the essential things are there, including glucose, amino acids, right? They all are there. So it is important. It is important to reabsorb. It is important to reabsorb. That's why the second step is tubular reabsorption. It is important to reabsorb the essential things. Are you getting my point? It is important to reabsorb the essential things. Are you getting my point? So as I said, there are certain pressures which will favor this filtration here. So they are written in front of you. They are not given in NCERT, but yeah, for the basic understanding of the topic, you need to know that. Right? You need to know that. Is that clear? Done? Okay. Done? So we have the glomerular hydrostatic pressure, we have the blood colloidal osmotic pressure, we have the capsular hydrostatic pressure. Is that clear? We have the glomerular hydrostatic pressure, we have the blood colloidal osmotic pressure, we have the blood colloidal osmotic pressure, we have the capsular hydrostatic pressure. We have the capsular hydrostatic pressure. Is that clear? We have the capsular hydrostatic pressure. Is that clear? Is that clear? Okay. So that's what you need to keep in your mind. Blood colloidal osmotic pressure of blood, and the capsular hydrostatic pressure. These are the three things that you need to keep in your mind. Okay. Okay. So this capsular hydrostatic pressure is due to the difference also. Just say afferent arteriole is narrow. So pressure will create here. That's how the filtration will occur. Now, bache, blood colloidal osmotic pressure. Now see, in the blood also, you know that the blood which is in the glomerulus, they are also having certain proteins in that. They are also exerting that osmotic pressure. They will also be like, nahi yaar, no, filtration should not occur. Then the filtrate present in the lumen of the woman's capsule will also be like, no, filtration should not be there. So here you people can see glomerular hydrostatic pressure is the blood pressure, right? Glomerular hydrostatic pressure is the blood pressure in glomerular capillaries due to the difference in diameter. Okay? It is the main driving force of filtration. This is important. It is the main driving force of filtration. This is important, right, bache? This is important. It is what? It is the main driving force of filtration. It is the main driving force of filtration. One second. Okay. Understood? So just keep it in your mind. This is the main driving force. So this pressure in that capillaries is important. This pressure in that capillaries is important. Done, bache? Next is the colloidal osmotic pressure. Next is what? Next is the colloidal osmotic pressure. Right, bache? Right. Right, bache? Next is the colloidal osmotic pressure. So when you talk about the colloidal osmotic pressure, it is the osmotic pressure created in the blood of glomerular capillaries due to the plasma proteins. So in the glomerular capillaries, the plasma proteins are present, and that are going to do that. Okay. Okay. Done? Okay. So it is the osmotic pressure which is created in the blood of glomerular capillaries due to the plasma proteins. So it resists the filtration of fluid from the capillary. Matlab, it is a pressure which is not favoring the filtration. It is the pressure which is not favoring the filtration. It is the pressure, right, which is not favoring the filtration. It is resisting it. Then capsular hydrostatic pressure, which is caused by the filtrate, that is there into the woman's capsule. So it resists the filtration. So it is about 10 to 18 mmHg. So net, the total effective filtration pressure will be, the net effective filtration pressure will be, see, this one is favoring filtration, these two are not. So we have to add it and subtract it. That's the value that we are going to get. That's the value that we are going to get. That's it. So let's talk about the reabsorption. So students, just keep it in your mind. The maximum reabsorption is there in the PCT, proximal convoluted tubule. Just for your reference, right? Just for your reference, I'm discussing the things from this diagram. This, you know that glomerulus and woman's capsule together, they form the renal capsule or Malpighian body, yeah, Malpighian capsule. Okay. So it is the renal capsule or Malpighian capsule. It is the renal capsule or Malpighian capsule. Right, it is the renal capsule or the Malpighian capsule. Right, bache? So glomerulus plus, again I'm repeating. So glomerulus plus, right? Right, what is there? Glomerulus plus. Glomerulus plus renal capsule, woman's capsule. Together they form Malpighian body or renal capsule. Renal capsule. That's what we have. Okay, that is what we have. Okay. So here we have the PCT, proximal convoluted tubule, where the maximum reabsorption is there. Right. You people can see bicarbonates are getting reabsorbed. NaCl, H2O, nutrients, and K positive. So here, if required, active transport is also there. Passive transport is also there. Passively, matlab no expenditure of energy. Your chloride ions, your water, it will be reabsorbed passively. But your glucose, your amino acid, right, they all be absorbed actively. Now this is the loop of Henle. This is the loop of Henle. I believe you know that. You know that the thin limb here, that's a PYQ, a very important PYQ, people. That's a very important PYQ. Please keep it in your mind. Okay, it's a very important PYQ, right? So here, what's the point, bache? This is the, you know that thick limb and thin limb. This is the descending limb. It is descending from cortex into the medulla. That's how you remember. So it is basically descending, right, bache? So here, what is going to happen? Here, what is going to happen? Yes, my dear people. So you know that this side is permeable to water, right? This side is permeable to water, but impermeable to electrolytes. Permeable to water, but impermeable to electrolytes. Permeable to water, but impermeable to electrolytes. Is that clear? Right? Is that clear? Is that clear? And this side, it is impermeable to, this is permeable to water, impermeable to electrolytes. And this side, yeah, solutes basically. So this side is permeable to solutes, but impermeable to water. See? And remember, from the thin side, this NaCl which is coming out, but this is moving out passively, right? Passively. But here it will be done actively. Here it will be done actively. Okay, here it will be done actively. Now it is the distal convoluted tubule. Here also you can see NaCl, water, bicarbonate, they all are what? Reabsorbed. And when you will see the arrow like this, right? Because in the text or in here in the diagram, this is marked differently. H positive is entering, matlab it is getting secreted. NH3A is getting added here, matlab tubular secretion it is showing. K positive is getting added here. H positive is getting added here. So here the water will be reabsorbed. This is all about this part, right? It is all about this part. Clear? It is all about this part. Clear? Clear, clear, clear. Sure? Sure? Right? So the same information is given here, right? I can start with the PCT also because I should start because the question will come. And this is the nephrons diagram. I believe there is no doubt here. I believe there is no doubt here. Okay? Baki, yes, see, no doubt in the NCERT. You know that when we say urine formation, we say it is just one topic. But you know that from single topic, 2 to 3 questions can come. Right? From single topic, 2 to 3 questions can come. Okay? 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 right? So in our case, in biology's case, it's not like single topic. From that single topic, there are many topics. Single topic is mitosis, many questions can frame. Yeah, single topic is cell cycle, one individual question. Mitosis say one question. Meiosis say 2 to 3 questions can come. So in our case, it works like that. Okay, in our case, it works like that. You know it. That's how we have to read, we have to study in the biology, right? That's how we have to read, we have to study in the biology. Is that clear, people? Sure? Sure? Done? 1 second. Okay? Done? So the first step here is glomerular filtration. So on an average, 1100 to 1200 ml of blood is filtered out by kidneys per minute. And this is the one-fifth of the blood pumped by each ventricle. Matlab here they are associating the topic with the cardiac output. And that cardiac output is also another MCQ, another important MCQ. So in biology's case, you know that from the theory itself, so many MCQs will come, right? So many MCQs will come. Okay. Okay, so basically cardiac output, right, bache? In the case of cardiac, how do we check it? Heart rate into stroke volume. How do we check it? Heart rate into stroke volume. So when you talk about the heart rate, yes, when you talk about the heart rate, just say on an average, we say 70 or 72. Let's consider it 70. And stroke volume, it is the amount of blood pumped by each ventricle in per beat. Okay, it is the amount of blood pumped by each ventricle per beat. So per beat it will be 70 ml. So it will be near about 5040 ml, roughly 5 liter. Basically, you can say 5 liter it is. So that 1100 to 1200 ml, it is basically 1/5 of. It is basically 1/5 of that cardiac output. So what is this cardiac output, people? What is this cardiac output, people? It's simple, right? It's simple that it is the amount of blood. So it is the blood pumped by each ventricle in a minute. So in a minute, matlab, how many beats will be there? In a minute, matlab, how many beats will be there? Done? Sure? Okay, chaliye, let's get started. So I believe this part is clear. So on an average, 1100 to 1200 ml of blood is filtered by kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of heart in a minute, right? So I hope this part is clear. So cardiac output is another topic, right? It is another topic, okay? Cardiac output is another topic that you need to mark. Done, bache? Chaliye. Okay, so glomerular capillary blood pressure causes filtration of blood. These 3 layers are already discussed. The filtration pressure is already discussed. Okay, bache. Right? Okay, so the epithelial cells of women's capsule, they are called poldocyte. This is the part that we have already covered. Now, this is the another important thing and another individual topic, that is GFR. Okay, that is what? That is GFR. That is what? That is GFR. This is the glomerular filtration rate. What's that? It is the glomerular filtration rate. What's that? It is the glomerular filtration rate. Please keep it in your mind, right? Please keep it in your mind. It is the glomerular filtration rate. Okay? So it is the amount of filtrate formed by kidneys per minute. In single minute, this is the amount of filtrate kidneys will form. In single minute, we are talking. So it is 125 ml per minute. Yes. So it is 125 ml per minute and it is 180 liter per day. It is 125 ml per minute and it is 180 liter per day. Is that clear? 125 ml per minute and it is 180 liter per day. Is that clear? Is that clear? Okay. So, this paragraph I'll discuss later. Baki, see, when you talk about the tubular reabsorption, as I said already, right? See, the filtrate formed is 180 liter per day. The filtrate formed is 180 liter per day, but the urine output is 1.5 liter. But the urine output is 1.5 liter, right? But the urine output is 1.5 liter. Is that clear? Is that clear? Is that clear? Done? Done, done, done. Sure. Any doubt here? Any doubt here? So this is the reabsorption. So you guys can see 180 liter of the filtrate will form and it is just the 1.5 liter. It is just 1.5 liter, that is the urine output. So nearly 99% of the filtrate will be reabsorbed. Nearly 99% of the filtrate will be reabsorbed. Okay. So tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism, right? Just for an example, glucose, amino acid, sodium ions in the filtrate, they are reabsorbed actively, right? They are reabsorbed actively. Matlab active transport is there against the concentration gradient, and nitrogenous base like your urea by passive transport, right? It will be done by passive transport. So reabsorption will occur passively in the initial segments of the nephron. It will occur passively in the initial segment of the nephron. Okay? And then what do we have? We have during urine formation, the tubular cells, they secrete substances like H positive, K positive, ammonia into the filtrate. So that is what you have to remember. And why is it done? It is done to maintain the acid-base balance. What is और? हाँ. ठीक? I'll show you something. He's such a cutie. See? Say hi. Hello बोल दो एक बार. She's Wasim sir's niece and she's so cute ना. She doesn't look like him. She's so cute. आप कैसे हो? आपको मैं अच्छी लगती हूँ? ये मामू डांटते हैं? थोड़ा सा तो डांटते. Yes, देखा? He is dangerous. चॉकलेट खाई? चॉकलेट खाई?
मामू से डर लगता है?
अभी नहीं खाना है. क्यों?
चॉकलेट किसने लाया?
What's my name?
नाम क्या है इनका?
अंबिका मैम.
है ना?
अंबिका।
तो चॉकलेट खाया ना? मैं कल को और लेके आऊंगी. ठीक?
अभी
नहीं खाना है. क्यों? देखा, she's so nice ना. She's so cute. She. है ना? और क्या पसंद है? चिप्स पसंद है? चिप्स भी लेके आओ? ठीक, मैं लेके आऊंगी. कोल्ड ड्रिंक भी लेके आऊंगी.
नहीं खाया कर.
Oh, doctor
has said
no to her. No
to chips.
No to chips.
Okay.
It's okay.
पढ़ाने तो मैम पढ़ा रही है अभी. मैम पढ़ा. ठीक है? चलो.
Bye.
बाय करो.
Bye करो.
बाय करो. बाय. बाय.
बाय. मैं जल्दी आऊंगी, नीचे बात करेंगे. She's so cute ना. She's so cute. She doesn't look like him. She's so cute. मतलब Wasim sir's sister, she's also very cute. But I don't know what happened to him. She's very nice. And she is just like an ATP for me. Chalo. So now come back to this. So tubular secretion. In the secretion, basically see, absorption mein we have kept whatever is required in our body. We have kept whatever is required in our body. And in the case of secretion, right, bache? So because it is to maintain the H positive balance, right? Ionic and acid base balance. It is to maintain the H positive, K positive, so ionic base balance. So that's why H positive, K positive ammonia, as per the need, they'll be added back. As per the need, they'll be added back. Okay, she, Radhya, she's so cute, na? I simply love her. So when I met her for the first time, she's like, Ambika ma'am. So she knows my name. Okay, so let's discuss that. Let's talk about the Henle's loop. So you know that reabsorption is minimum in the ascending limb. So it is maximum in bache. It is maximum in reabsorption is minimum in the ascending limb. Where is reabsorption minimum? Right? Tell me. Tell me. Yes, so in there, right? So there it is minimum. Is that clear? Is that clear? Is that clear? Sure. Chalo, let's continue. Okay, let's continue. So reabsorption is minimum in the ascending limb. However, this region plays a significant role in the maintaining of high osmolarity of medullary interstitial fluid. Why do we say so? Because when you talk about the descending limb, as I said, in the case of descending limb, it is permeable to water, but impermeable to these NaCl and all, to these ions or solutes, whatever you want to say. Here, this is permeable to this. So of course, right, from the thin part passively, from thick part actively, right? This NaCl will enter here in the medullary fluid, right? You know that in this loop of Henle is there in the medulla. In kidney, we have cortex, we have medulla. Medulla is having the medullary fluid. So we have this thing here, right? We have this thing here. So NaCl will be added here. So of course, it is increasing the osmolarity. That is also required. Why it is required, bache? Because we have to reabsorb the water from this side as much as we can. And for that particular thing, my dear students, right, bache? Yes, we have to maintain the osmolarity here. Okay, we will discuss it in countercurrent mechanism. Okay, we can discuss it in countercurrent mechanism. So here also DCT, from DCT, there is the conditional reabsorption, right? From DCT, there is the conditional reabsorption as per the need, right? As per the need, the water will be reabsorbed, sodium ions will be reabsorbed, right? And here also. Chalo, one by one, let's discuss that. Take, so Henle's loop mein, I told you the descending limb of the loop of Henle is permeable to water, but impermeable to electrolytes. Take, so this concentrates the filtrate as it moves down. Am I right? So descending limb is permeable to water. Of course, when that filtrate is going down, water is coming out of it. So of course, if water is coming out of it, more and more solute is in it. So it means the filtrate will become concentrated. What's that? That's a PYQ. What's that? That's a PYQ. What's that? That's a PYQ. Any doubt? Any doubt? Any doubt? Any doubt? Sure? Sure? Okay. So this is a PYQ. Now when the filtrate is going up in the ascending limb, now electrolytes are coming out. Mane solute is coming out. So of course, that concentrated filtrate will become diluted again, right? So the ascending limb is impermeable to water, but it will allow the transport of electrolytes actively or passively, right? So the ascending limb is impermeable to water, but it allows the transport of electrolytes actively or passively. So as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolytes to the medullary fluid, right? It gets diluted due to the passage of electrolytes to the medullary fluid. Any doubt here? Any doubt here, people? This is important. Any doubt here? Yes? Chaliye. So next is collecting duct again, right? This will pass from the cortex to the kidney. This will pass from the cortex of the kidney to the inner parts of the medulla, of course, and it is for the water reabsorption. It is for the water reabsorption. So large amount of water would be reabsorbed from this region to produce a concentrated urine. Large amount of water would be reabsorbed, right, from this region to produce a concentrated urine. So this segment will allow the passage of small amount of urea as well. So when I will teach you the countercurrent mechanism, then you will understand this. So up to this part, all clear, right? Which limb is permeable to what? That clear? Because next is countercurrent mechanism, then RAAS mechanism, then we'll shift to the respiratory volumes and capacity, mechanism of breathing, and exchange of gases वाला part भी कर लेंगे. चलिए, हाँ जी, doubt? Hello, doubt? देखो students, you already know that I'm taking the lecture from somebody's studio, matlab your Wasim sir's studio. I have to go home as well. So if you are interested in completing it, tell me. If you people are not interested in completing it, I'll take it in another lecture. It's all up to you. But do answer my question. Do answer my question. Doubt hai, yeah, if there is something which you are not able to understand, yeah, if there is something which is bothering you, right? So if you think madam will start from the basics, I'm doing that. But if like you don't know anything from that chapter, I would suggest you to watch our Game of NEET 2.0 or something like that. End game, something like that. End game. You guys can watch. Right? Geeta bhi in countercurrent mechanism, we'll discuss that. Okay. So guys, how's the Josh? How's the energy? How's the Josh? How's the energy? How's the Josh? How's the energy? High or low? What's that? High or low? Great. Right? What about others? What about others? Arey, it's okay, fine. Whosoever is chatting there, leave them. Done? Okay, chali. So now let's talk about the countercurrent mechanism, right? And I know many students, they find it difficult. So I'll try to make it easy for all of you. This is the kidney that we have, right? This is the kidney that we have. So this one is the outer cortex. This one is the inner medulla. This one is the outer cortex. This one is the inner medulla. Now, when you look at this part, now, when you look at this part, so this is cortex, this is medulla. Here you have medullary fluid or the interstitial fluid. Medullary fluid or the interstitial fluid. So you know that the things are placed like this. The things are placed like this. That's how it is. Done. That's how the things are. So remember one thing. Remember one thing. So this is what cortex is. This is what medulla is. So basically, there are two type of nephrons that we discussed. One is cortical nephron, another is juxtamedullary nephron. One is cortical nephron and another is juxtamedullary nephron. These are the two things that we have. The cortical nephron and the juxtamedullary nephron. The cortical nephron and the juxtamedullary nephron. So when you talk about the cortical nephron, okay? So in their case, the loop of Henle, bache, it's quite short, right? Even this, when you will look at the woman's capsule PCT, they are near to the surface. Just let's suppose if it is the cortex, here you will see this U-shaped loop of Henle is not that deep. This U-shaped loop of Henle is not that deep. Keep it in your mind. But in the juxtamedullary nephron, so they are making near about 10 to 15% of the nephron. They are making near about 10 to 15% of the nephron. So in their case, they are making near about 10 to 15% of the nephron. This juxtamedullary nephron, right? This juxtamedullary nephron, they are making 10 to, yes, bache, right? 10 to 15% of the nephron. In their case, you will see that the loop of Henle is quite long. Loop of Henle is quite long. It is very long. It is deeply there in the medulla. It is deeply embedded there in the medulla. It is deeply embedded there in the medulla. Are you getting my point? And this one, because this is also covered with vasa recta. Do you know what vasa recta is? Do you know what vasa recta is? Let's suppose this is your loop of Henle. Let's suppose this is your loop of Henle. Now, as I said, it is covered with vasa recta. That is the capillary network. Vasa recta. That is the capillary network. What's that? That is the capillary network. 1 minute. See, this is what we have. This is what we have. So this is the loop of Henle. This is the loop of Henle. Is that clear? This is loop of Henle. This is the descending limb of loop of Henle going down. This is the ascending limb of loop of Henle going up. Now it is covered with vasa recta. It is a network of blood capillary. Now always remember, that's something that you need to remember. Descending limb is always having ascending limb here. Ascending limb is always having descending limb here. That's what you need to keep in your mind. That's what you need to keep in your mind. Okay. That is what you need to keep in your mind. Is that clear? Is that clear? Is that clear? So this is the arrangement that we see actually. This is actually the arrangement that we see. This is the descending limb of loop of Henle. This is the ascending limb of vasa recta. And when you look at this, bache, see, in the descending limb, filtrate is going down. In the ascending limb, filtrate is going up. So it is the, like, what is it? It is the counter current. Like, they both are in the opposite direction. That's why we use the word counter current here. Right? Right? Yes or no? Yes or no? They both are in the counter current. Yes or no? Yes? Yes or no? Yes. So descending limb is going down, ascending limb is going up. So this is the loop of Henle that we have, right? Here the filtrate is in the opposite direction, number one. Here you have the vasa recta. Here what do you have? Here you have the vasa recta. In the case of vasa recta, right, bache? In the case of vasa recta, right? In the case of vasa recta, you will see this is the descending limb where the blood is going down. This is the ascending limb where the blood is going up. So now when you look at these two things, descending limb of loop of Henle is having ascending limb of vasa recta, right? So here when you will see, they both are in opposite direction. So here also you will see the counter current. Here also you will see the counter current. Here also you will see the counter current. Here also the filtrate is going up, the blood is going down. They both are also in opposite direction. So counter current mane, this is in opposite direction. So this is the general arrangement. This is the arrangement that you need to focus. This is the arrangement that you need to understand. So this part clear, people? Sure? This part clear? Sure? Are you sure? Are you sure? Okay. Okay. ठीक, चलिए. So now, right, in the surrounding, what do we have here? Matlab, this is your loop of Henle. This is your vasa recta. In the surrounding, of course, we have the medullary fluid. Yes, we have the medullary fluid. Always remember that it is the NaCl and it is the urea that maintain osmolarity of the medullary fluid. That is also a PYQ, right? This is the NaCl and urea that maintain osmolarity of medullary fluid. These are the one that maintain osmolarity of medullary fluid. That is also an important question. That is also an important question. Now, what's the point here, right? You know that this side will give water, this side will give NaCl. Am I right? This side will give water, this side will give NaCl. Right now, before explaining you the topic, right now, before explaining you the topic, I'm expecting you people to remember a few things, right? I'm expecting you people to remember a few things. Then everything will be easy for you. So as I said, so from this side, water is coming down, from the, water is coming from this side, NaCl is coming. Now, this is the descending limb. In setting, he's so this is the descending limb. This is the ascending limb. This is the descending limb. This is the ascending limb. This is the descending limb. This is the ascending limb. And this is the ascending limb, right? This is the descending limb, right? And this is the ascending limb of vasa recta, that is blood capillary. So now what's happening? Your descending limb of loop of Henle is, right? This descending limb is permeable to water, impermeable to electrolyte. This one is permeable to electrolyte, impermeable to water. Now, this ascending limb, when you talk about this ascending limb, right, this is the blood capillary. It is like I am okay with everything, right? I have no problem if water is coming from whatever side. If water wants to come from any side, it can come. Electrolytes wants to come, it can come. Now, this setup is basically to conserve the water, because in the birds, in the mammals, there is the capability that they can, right, make the filtrate 4 times more concentrated. Whether the urine is 4 times more concentrated than the filtrate, right? So how is it possible? With the help of this countercurrent mechanism. So as I said, water is coming out from this side, NaCl. You remember it in this way. Descending limb is giving the water to the medullary fluid. Ascending limb is picking it up and it is giving out NaCl. Ascending limb is giving out NaCl and medullary fluid. Descending limb is giving up, picking it up and giving out water. Actually, that is the arrangement that you need to remember, right? Actually, that is the arrangement which you people need to remember. So again, I'm repeating this. The descending limb of loop of Henle is throwing out water in the medullary fluid that will be picked up by the ascending limb of vasa recta, right? And meanwhile, it will throw NaCl. Now, here, ascending limb will throw out the NaCl here. That will be picked up by descending limb and it will give out water. This is something you need to remember. Then I'll explain you the logic. Right? Then I'll explain you the logic. Matlab, it's like, jase Adiba was there, na, kids. Sometimes kids, they pick up your stuff. You want them. So you'll give them some other distraction and they'll give it back to you. They'll give it back to you. Same is the thing here. Here, water is there in the surrounding. This ascending limb of vasa recta will pick it up, will throw out NaCl. Here, it will pick up NaCl, will give out, DL will pick it up, will give out water. So firstly, remember that. Firstly, remember that. Now, how things are going on? How things are going on? Here you have the cortex, here you have the medulla. Here you have the cortex, here you have the medulla. Okay? So when you move from cortex to medulla, to the inner side of medulla, you'll see osmolarity will change. I said 200, 300, slowly 400, 500, and finally it will reach to 1200. So it is m osmol per liter. This is the unit that we use. It is m osmol per liter. This is the unit that we use. It is the m osmol per liter. That is the unit that we use. Up to this part, all clear? First of all, tell me. Yes? Tell me. Tell me. Up to this part, all clear? Up to this part, all clear? Sure? Sure, sure, sure. Up to this part, all clear? Good. So, osmolarity, when you move from cortex, outer cortex to inner medulla, deep down, the osmolarity will increase, right? The osmolarity will increase. Is that clear? The osmolarity will increase. That's what you need to keep in your mind. So, it is obvious now, like let's suppose this is the loop of Henle, right? This is the loop of Henle. And as you know, in the surrounding vasa recta is there. But it's simple. You know that ascending limb is playing no role in the, from the ascending limb, basically minimum reabsorption is there, but the electrolytes are coming in the surrounding. So these electrolytes, what will they do? Right? It is all the concept of osmosis. If you understand osmosis, you know that there are two cells, right? Osmosis is the movement of solvent from, right? It is the movement of solvent from its higher concentration to low concentration through a semi-permeable membrane. What is osmosis? It is the movement of solvent, yeah, movement of water from its higher concentration to lower concentration, from its higher concentration to lower concentration, right? Right, through the semi-permeable membrane. Now, let's say if this is the cell having more solute, less solvent, and this is the cell in comparison to this having more solvent, less solute, definitely this is B, this is A. Water will move from B to A. Water will move from B to A. The water will move from B to A. Is that clear? Is that clear? And ultimately in the body, the conditions has to be isotonic. Matlab, isotonic conditions will be maintained. Right? Now here what is happening, from this ascending limb, electrolytes are coming here in the medullary fluid. So they will increase the solute concentration there. They will increase the osmolarity there. So that is why from this side, more and more water will come out, because it has to balance it out. That's the main agenda actually. Right? That is the main agenda actually. So it has to balance it out. Right? It has to balance it out. Are you getting my point? It has to balance it out. Are you getting my point? Are you getting my point? So basically what is happening, electrolytes are coming from this side. They are in the medullary fluid, and from this side, right? And from this side, what is happening? Because more and more solute here, the water has to come out. Water has to come out, because if solute concentration will be more, right, and here if it will be more low solvent, that will eventually come out, and then that water will be picked up by the ascending limb of vasa recta, and it will be passed to our body, right? Matlab, it will increase the body fluid volume, right? And that is what we need. This water has to take out, because we have to. This is what we need. We have to absorb more and more water, more and more water, more and more water. That is the first thing. Now, when this side is picking up the NaCl, it is also giving some amount of water now, right? Because here it will be otherwise too much of osmolarity. It is giving some amount of water, but majorly from this side, we have to take as much water as we can, so that water will further enter in our body circulation. That is how the things are. So that's why, because water is coming out. So when you move the descending limb, when you move down the axis, so you will see osmolarity 400, 500, 600, 800, 1000, 1200. So when it will reach here, it will be highly, right, concentrated, right? It will be highly concentrated. Is that clear? Is that clear? Keep it in your mind. Okay, keep it in your mind, please. Okay, now the another thing is, from this side is picking up water, it is also adding the NaCl. So if this side of the vasa recta is picking up NaCl, this side is giving the NaCl back. This side is giving the NaCl back. This side is giving the NaCl back. So NaCl is not going anywhere. Ultimately, it is there in the medullary fluid only, because it has to maintain the osmolarity. Because it has to maintain the osmolarity. Is that clear? Is that clear? Is that clear? And same is the case with the collecting duct also. When here there will be the more solute, the water will be taken out here. And always remember, some amount of urea, right? It will be reabsorbed from collecting duct. It will be added here. It will be added to the filtrate. So that is also maintaining the osmolarity. That is also maintaining the osmolarity. Is that clear? That is also maintaining the osmolarity. Is that clear? Is that clear? Okay. Done? Done? Any doubt? Chale, well done. So that's how the things are, right? And I hope now this part is clear. I hope now this part is clear. Ye dekho, right? From this collecting duct, the urea will be reabsorbed. It will be added back to the filtrate, right? It will be added back to the filtrate. So that is how it is. The mechanism of the filtrate. So do read about it. Whatever points I have explained, whatever is written in NCERT, that is basically discussed here. So see, from this side, water is coming out, NaCl is coming out, water is coming out. So from collecting duct also, the water will be reabsorbed because this side is adding more and more electrolytes here, right? So the diagrams are here. They will make this topic easy. So just observe it once. Just read about it once. Yes, just observe it once. Just read about it once. Quick. 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 Done? So that's it. Next is the regulation of kidney function. So you know that juxtaglomerular apparatus will help here. So if you don't know juxtaglomerular apparatus, just quickly I'm telling you, it is having the juxtaglomerular cells, also known as granular cells, and we talk about the macula densa. I'll show you the diagram. Ye dekho. So when you look at this part, na, this JGA, this juxtaglomerular apparatus, something very important. Because this juxtaglomerular apparatus, bache, it maintains the GFR. It maintains the glomerular filtration rate. It maintains the GFR. It maintains the glomerular filtration rate. What will it do, bache? What will it do? It will maintain the glomerular filtration rate. So when you look at this, these are the atrioles here. These are the atrioles here. Right. So the nephrons are arranged in such a way. So when the DCT, when this DCT it comes in contact with, when the DCT it comes in contact with these arteriole, particularly the efferent arteriole, the cells of the DCT will get modified. And what do we call them? We call them as the macula densa of distal tubule. What do we call them? We call them as the macula densa of distal tubule. We call them as the macula densa of distal tubule. Is that clear? Is that clear? Plus here what you people will see. Plus here, juxtaglomerular cells, right? They are in the wall of this arteriole here. They are also known as granular cells. They are also known as renin. They are also known as granular cells. They are also known as renin. This is what you need to keep in your mind. So this juxtaglomerular apparatus, whenever bache, the GFR is low, right? Glomerular filtration rate is low. So what are the reasons for the low GFR? What are the reasons for the low GFR? It can be anything. It can be low blood pressure, low blood fluid volume concept. It's like that. So basically your JG cells, they release renin. It is RENIN, single N, right? Double N wala renin is an enzyme, right? Renin, that is the activated form. That is a proteolytic enzyme that is there in the digestive secretion, gastric secretion to be more precise. So JG cells, when the GFR is low, these JG cells, they release what? They release the renin. They release the renin. Now what's going to happen? Now what's going to happen? In the blood, in the plasma, you have a plasma protein, angiotensinogen. Angiotensinogen. It will act on this, renin will act on this angiotensinogen, and that will form angiotensin 1, and then it will form angiotensin 2. Matlab, this angiotensinogen is the inactivated form, right? This is the inactivated form. This is the inactivated protein, okay? This is the inactivated protein, and it is synthesized by liver. It is synthesized by liver. Who's going to synthesize it? It is synthesized by liver. Am I clear? It is synthesized by liver. So angiotensin 1 to 2, ACE. What is this? It is angiotensin converting enzyme. It is angiotensin converting enzyme. What's that? It is angiotensin converting enzyme, right? So what do we have here? Angiotensin 2. Now, remember, this angiotensin 2 will also act on PCT, that is proximal convoluted tubule, will basically, it will also act on that angiotensin 2, right, bache? So why? So what will it do when it will act on that Sorry, angiotensin 2 will also act on PCT. So when it will act on PCT, something is going to happen. Can you tell me about that? See, this is the topic that we are discussing is RAAS mechanism. The topic that we are discussing is RAAS mechanism, renin, angiotensin, aldosterone system, right? Even if you want to see a short video on that, you can watch it on Unacademy NEET English channel. I have already uploaded that. Now, what am I saying? That angiotensin 1, it will get converted, it form angiotensin 2. And I'm saying it will act on PCT. And when it will act on PCT, it will increase blood pressure, it will increase body fluid volume, it will also play a role in increasing the GFR in this way, but it will act on PCT. So will you please tell me what will it do? What will it do? Right, it acts on PCT, but it is also having the vasoconstrictory effect, right? It is also having that vasoconstrictory effect. It will act on that. It will act on that proximal convoluted tubule. The vasoconstriction will be, right? So vasoconstrictory effect is there. So it will also act on PCT. First of all, mark this. First of all, write this, right? First of all, write this. Quick. Yes, quick. Yes, please. Yes, please. So what will it do here? What will it do here? When it will act on PCT? When it will act on PCT, anybody? I just said that it will increase the GFR. How? Because ionic absorption. जैसे मान लो sodium ion का, even bicarbonate ion, HCO3 negative, it will play the role in their reabsorption as well. Right, bache? Right, bache? So further, this angiotensin 2, it will act on the adrenal gland. And to be more precise, adrenal cortex. Adrenal gland, you understand now? Cap of kidney. So when it will act on adrenal cortex, right, that cortex part will release aldosterone. And this aldosterone, my dear students, it will act on your distal convoluted tubule or the collecting duct, means on the later part of the nephron. This one will be on the proximal part. This one will be on the distal part, right, bache? So again, there will be the conditional reabsorption of sodium of bicarbonate. I have already discussed that, right, bache? So this is the RAAS mechanism and it is required to regulate the GFR. It is there to regulate the GFR. It is there to regulate the GFR. ये देखो. So renin will release from kidney. Who's going to release that? JG cell, juxtaglomerular cell. JG cell, juxtaglomerular cell. Right, bache? So angiotensinogen will be converted to angiotensin 1 and then to 2. So AC is the enzyme that will help in their conversion, and this is released from lungs. This is released from lungs. And this angiotensin 2 will also act on blood vessels. I told you it is having the vasoconstrictory effect. It constricts the blood vessel, definitely will increase the pressure. But do remember, it also act on PCT. And it will act on the adrenal cortex. Aldosterone will be released. That will act on the kidney. And then there is the reabsorption of sodium, ion, and water, basically, NaCl and water. Right? NaCl and water. So there is a homework. If let's say in our body and we have to reabsorb sodium, right? Then what has to be excreted out? That's your homework. What has to be excreted out? That's your homework. So RAAS mechanism, it's a very important topic. And remember, your ANF, that is your atrial natriuretic factor. That is your atrial natriuretic factor. That is your atrial natriuretic factor released by heart. So it acts like a check. It acts like a check. It acts like a check on. Yes, my dear people, it acts like a check on. It acts like a check on RAAS mechanism. Again, a direct PYQ. Again, a direct PYQ. Done? Chaliye, let's move to the next part. One question will come from the respiratory volumes and the capacity. So it is the, yes, it is the pyrometer that will help in there, that we can use to check it. Okay, bache, to diagnose it. Okay. Chaliye, then let's go. Let's discuss that. So tidal volume, right, bache? Tidal volume, inspiratory reserve volume, expiratory reserve volume, and residual volume. Right? Let's talk about that. So who's going to help? Which one will help to check it? It is the spirometer. It is the spirometer. It is the spirometer. Done? Sure? Are you sure? Are you sure? Chali, let's discuss that. So spirometer will help to check it. But yeah, remember residual volume will not be checked. Okay, so overall spiral volume. So tidal volume, inspiratory reserve volume, expiratory reserve volume. So TV, IRV, ERV and RV. So what is this tidal volume? Just say without any extra efforts, right? Even if I'm talking to you, I'm inhaling and I'm exhaling. That is tidal volume, right? And per breath, it is around 500 ml. Okay, so on an average, in the healthy individual, you will say 12 to 6 breath per minute. It will be 12 to 16 breath per minute. 12 to 16 breath per minute. So volume of air inspired or expired during a normal respiration. Volume of air inspired or expired during a normal respiration. So it is 500 ml. Tidal volume is 500 ml. So out of this 500 ml, bache, right, you can say 350 ml will be there for the exchange and 150 ml, right? 150 ml will make the dead space volume, right? It is not used for the exchange there. Right, it is not at all used for the exchange there. So now I'm not entering in the depth of this topic. So simple 500 ml, and as I said, so approximately 6,000 to 8,000 ml of air per minute, because see now 12 to 16 time per minute. So 12 into 500 and 16 into 500. 12 into 500, 16 into 500. That's how you can remember. Okay, that's how you can remember. Yes, 150 is the dead space volume. Right, 150 is the dead space volume, not available for the exchange, but 350 ml is available for the exchange. And why is it so? Do let me know in my regular lecture, right? In my detailed lecture, I have already discussed that. Inspiratory reserve volume, something is related to inhalation, inspiration. So additional volume of air, a person can inspire. Additional volume of air, a person can inspire by a forcible inspiration. Additional volume of air, a person can inspire, just normally. Now, additional volume of air, a person can inspire. Are you getting my point? Are you getting my point? Right? So this is the inspiratory reserve volume on an average. For each breath, it is 2,500 ml to 3,000 ml. 2,500 ml to 3,000 ml. Then it comes the expiratory reserve volume. So the person can expire, forcible expiration. Right, bache? 1,000 ml to 1,100 ml. Yes, it is important to remember. Residual. Our lungs will not always be empty, right? There will be some amount of air in our lungs always, right? There will be some amount of air in our lungs always. Are you getting my point? There will be some amount of air in our lungs always. Just keep it in your mind, guys, right? Just keep it in your mind. So when you talk about that, right? So what is that? Yes, please. What is that? That is the residual volume remaining. It is 1,100 to 1,200, but expiratory reserve volume is 1,000 to 1,100. What's that? It is 1,000 to 1,100. So any doubt? Pakka? Right? Sure? Okay. So let's go for that. Let's discuss that. So residual volume, it is the volume of air remaining in the lungs even after the forcible expiration. So question will definitely come from this part. We have tidal volume, we have IRV, we have ERV, and we have residual volume, right? And we have residual volume, tidal volume, inspiratory reserve volume, expiratory reserve volume, and residual volume. So it is 6,000 to 8,000 ml, or per breath pe to it is 500 ml, you know that, right? And this one will be 2,500 to 3,000. Please correct me if I'm wrong. Yes, please correct me if I'm wrong. Please correct me if I'm wrong. Is it 2,500 to 3,000? Is it 2,500 to 3,000? Yes, please. Is it 2,500 to 3,000? Yes, please. 2,500 to 3,000. Batao. At least say yes or no. right? Now when you talk about the capacity, now when you talk about the pulmonary capacities, so capacities are they are basically, when you add the volumes, you will get the capacities, right? When you add the volumes, you will get the capacities. When you will add the volume, you will get the capacities. Matlab, capacities are the combination of the volume. Capacities are the combination of the volume. So with the help of spirometer, we cannot check residual volume. So if we cannot check residual volume, so the capacities where we calculate residual volume will not be checked as well. Keep it in your mind. So here you have a trick: inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, functional residual capacity. Inspiratory capacity, right? Expiratory capacity, vital capacity, total lung capacity, and functional residual capacity. Functional residual capacity, right? So inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, functional residual capacity. Please explain it in the chat. Please mention it in the chat. Please mention it in the chat. Yes. So inspiratory capacity, expiratory, vital, total lung capacity, and the functional residual capacity. So remember, you have to write total tidal volume, tidal volume, tidal volume. Go like this: tidal volume, tidal volume, tidal volume, tidal volume. Go like this: tidal volume, tidal volume, tidal volume, tidal volume. Now, if it is inspiratory capacity, add inspiratory reserve volume. If it is expiratory capacity, add expiratory reserve volume. Add expiratory reserve volume. If it is vital capacity, add inspiratory reserve volume and expiratory reserve volume. If it is total lung capacity, you have to consider everything. Then bache, you have to add residual volume here as well. You have to add residual volume here as well. So we also write it like this: vital capacity plus residual volume. We also like it like this: vital capacity plus residual volume. And when it is the functional residual capacity, so it is the residual volume plus ERV. Expiratory reserve volume. Expiratory reserve volume. Is that clear? Right? Is that clear? Is that clear? Sure? Are you sure, people? Are you sure? So inspiratory capacity, expiratory, vital, total lung capacity, and functional residual capacity. So here, this is what you have. So when you will add tidal volume plus inspiratory reserve volume, you'll get the value of this. So that's what you have to remember. So firstly, remember their values, then inspiratory, expiratory, vital, total, functional residual capacity. You have to write tidal volume, tidal volume, tidal volume here, but in functional residual capacity, R for residual volume, and because you are talking about the remaining part, you have to consider the ERV as well. Okay, and then here, that's how it is. When it is inspiratory capacity, add IRV. When it is expiratory capacity, add ERV. When it is vital capacity, add both. Right? Add both. When it is total lung capacity, add TV, IRV, ERV, residual volume. So that's how it works. Fine? That's how it works. So do we have any doubt here? Yes. Do you have any doubt here? Yes, people? Do you have any doubt here? If there is any doubt, please feel free to ask. If there is any doubt, feel free to ask. Okay, so this is all about the capacities. Then, bache, when it comes to the mechanism of breathing, right? Firstly, remember Boyle's law. Firstly, remember Boyle's law. And that says if the volume is high, the pressure will go down. If the volume is high, the pressure will go down. Number one, right? Number two, you have to remember which muscles are the inspiratory muscles. So the muscles which contract during inspiration, they are the inspiratory muscles, like your external intercostal muscles and your diaphragm muscles. So the question will come from the muscles part. Right, bache? So rest everything. Let's continue in the next lecture. Okay. Rest everything. Let's continue in the next lecture. So you know that basically we have 4 parts, right? 4 classes for each topic, no? 4 classes for each topic, no? Yes? So we will complete it then. So after this particular class, what will be our how are we going to complete our next lecture? As I said, I'll pick up 20 questions, right? What will I do? I will pick up 20 questions, 20 concepts, right? Let's say I'll pick up 20 concepts. So from each concept, if I will pick 2 questions, matlab 40 questions. So with the help of questions, we will solve it, right? We'll reverse the things. We'll change our way. Okay. So is it okay for all of you? If it is a yes, do let me know in the chat section. If it is okay for all of you, do let me know in the chat section. Do let me know in the chat section. If it is a yes, so if you are happy with the lecture, if you are happy with the quick revision, because I'll be picking up the most important topics, even from plant physiology, even from human physiology, we'll pick up the most important topic, even from genetics. So if it is a yes, if you are happy, if you like the session, so I want to see some fire in the chat section, right? I want to see some fire in the chat section, quick. Thank you, Bache Faisal. Thank you. Hello, Gayatri. Okay. So see you all soon, right? So hopefully not Sunday, because Sunday Yava sir is taking the class. So I'll take your class on Tuesday. Okay. On Tuesday, I'll be taking the class. Fine. Fine. Okay. So here I'm ending the lecture. Hope you liked it. And if there is any doubt, feel free to ask. Feel free to ask. Done. So if you like the session, do let me know in the comment section. Take care. Thank you so much, people. Bye-bye.